वो जिसने रजिस्टर किया उसको
तुष्णा मैडम फर्स्ट लेफ्ट में ऊपर नीले पे extreme left.
Can you hear me?
Tushna madam, you are there?
Yes, sir. Can you hear me?
Yes, madam. You are audible, madam.
Okay.
We will just start in 2 minutes, madam. We are waiting for some small things to.
Sure, sir. Sure. Sir, I'll share this screen and put up the PowerPoint if that's okay. Just check if it's working.
Madam, we can see the presentation.
Okay. Yeah.
Students there, yes, well. So good morning, students, and good morning, Tushna madam, and a very good morning to all other delegates, faculty members who have joined online. So today we have with us Dr. S.W. Savarkar sir. He associate professor and head department of extension veterinary extension, and sir will be acting as the chairman of the session. We have Dr. Pfeffer with us, who will be joining shortly. He is assistant professor in the department of veterinary pathology. And we also have Dr. Vivek sir with us. He is assistant professor in the department of veterinary biochemistry, and Dr. Jignesh Chawda, who is assistant professor in the department of veterinary animal nutrition. So now I hand over the session proceedings to the chairman of the session. Sir will briefly introduce Dr. Tushna, and thereafter Madam can start with her presentation.
Thank you, Dr. Elawan sir. Very good morning, Dr. Tushna madam. Very good morning, dear students, trainees and friends. So today is the third day of this 5 days training program. And there are very good lectures. First lecture on breeding for conservation of wild animals by Krishna Karkaria, who is a project veterinarian, Wildlife Institute of India, Dehradun. Second lecture will be captive breeding and reintroduction of endangered wildlife by Dr. P.H. Vatalia, Director Extension of Education, Kamdin University, Gandhinagar. Third lecture will be bird rescue and rehabilitation by Sri Yagnesh Bhatt. Fourth lecture will be challenges and opportunities in wildlife disease diagnosis by Dr. S.K. Panda. And fifth one will be rhino translocation, in particular as a tool for securing survival of the species. So Dear students, all 4 lectures are very good for your wildlife information. And now we will going to start first lecture of breeding for conservation of wild animals. You know that conservation is very important part of wildlife, and breeding is most important aspect for reproduction. Now regarding this today's speaker for this particular lecture, Dr. Krishna Karkaria, I will introduce her. Dr. Krishna graduated BVC and EH from Veterinary College Anand in 2015. She had completed a master in veterinary science, wildlife study from Kerala Veterinary and Animal Science University, Bokode, with research focus on screening for disease in Asiatic wild dog in South Wayanad Forest Division in Kerala. In 2018, she also completed integrated training program in infectious diseases, food safety, and public policy from University of in Canada during 2017. Dr. Krishna developed various skills in wildlife capture and in mobilization, wildlife rescue, and rehabilitation and management in ex situ conservation project. At present, she is working as a project veterinarian in Wildlife Institute of India, Dehradun, that is the national level institute. Since she is working since September 2017, and working as a lead veterinarian for conservation breeding project of Great Indian Bustard, and also practice veterinary medicine with emphasis on preventive care, husbandry, disease management, and emergency medicine for the bustard recovery project. So, dear students, you are having a very good veterinarian. She is having a good experience in wildlife area, and I think that her lecture and her information will definitely inspire you for getting your career, not only information in particular veterinary area, but also she will inspire you for doing a good career in wildlife area. So again, I welcome Dr. Krishna Karkaria, and I hand over this stage to Dr. Krishna. Dr. Krishna, please.
Good morning, everyone. As Sir introduced me, I am working with the Bustard Recovery Project with the Wildlife Institute of India, and I have been working on this project since 2017. And I'll be talking about conservation breeding, a brief on conservation breeding. So the why, who, and what of conservation breeding. So why is conservation important in this stage? So the conservation is basically protection, upliftment, and scientific management of biodiversity so as to maintain it in its threshold level and to derive sustainable benefits in the present and for the future generations. This definition from biodiversity and climate change adaptation from 2018 is very precise, and it summarizes the point of conservation. Given the current habitat fragmentation and its effect on animal population, species conservation over the long term increasingly require management, and there are two basic management strategies, that is the in situ and the ex situ conservation management. So in situ, what happens is that you manage the species in its natural habitat, that is in national parks, while ex situ management involves removing the species from its natural habitat and keeping it in captivity and using it in zoos for conservation. So conservation breeding is a type of ex situ management. There is a slight difference between captive breeding and conservation breeding. All species that are bred in captivity are captive bred animals. So in zoos and parks, there are species which are breeding, but if they are not used for conservation, it would not be considered conservation breeding. So the main difference between conservation and captive breeding is that the conservation breeding programs will focus on reintroducing the populations in the wild, and it is not for commercial or recreational purposes only. And according to IUCN, now that the populations are declining, any population below the number of 1,000 individuals should be considered for conservation breeding. But according to that, 752 species are less than 1,000 individuals and may require conservation breeding programs for them. So there's a huge task ahead of us, and we'll have to work with the conservation breeding facilities and in the field with conservationists to cooperate and make complementary efforts. So conservation breeding alone is not sufficient. There is a need to preserve the environment where the species will be released. And the ultimate goal of conservation breeding is to recreate a self-sustaining wild population. So there are different kinds of conservation breeding and breeding programs that are there. There are some which are necessary. That means that the population has been extinct in the wild. So there are only the ones which are kept in captivity and bred for future. So this picture on the top right is of the macaw. There are no species, there are no birds left in the wild. So there is only a captive population, and now they have reached the numbers to 150 from 8 individuals that started in captivity. Like this, there are many other species like the California condor, which we'll be studying in a little more detail. So they are extinct in the wild, so we have to breed them in captivity and get their numbers higher and then release them. Then there is the second is integral. That is, it is one of the many conservation tools. The picture on the bottom left is of the houbara bustard. The houbara bustard is a species of desert and grassland areas. And it is not extinct in the wild, but needs conservation breeding program along with other strategies that is implemented. There is the third kind, which is precautionary, which is a backup. So in gypsy vultures, in most of the Indian vultures, the numbers plummeted due to the rise in deaths due to diclofenac toxicity in the vultures. And since then, the conservation breeding program has started in India for getting the numbers of the vultures in captivity higher. So this would be as a precautionary or a backup step. The other is prudent, which means that there is already a captive population present, and so it is prudent to use this captive population to increase the numbers and release them in the wild. So in this, the cheer pheasant is an example in India where cheer pheasants were kept in captivity and then bred for future release. Then there is motivational kind of breeding programs. That is, it draws more attention, the iconic 3 species that are very enigmatic and capture attention. They are bred in captivity for conservation. So they will build support for conservation. And then there is market driven, that is meeting the trade demand. It does not mean that it is supplying the trade, but it is supplementing the species that is already in market. And the example for that would be the green avadavat. It is a cage bird species. There are different advantages of conservation breeding programs. The number one is it builds up a large number for re-establishing the population in the wild. The animal bred in conservation facilities can be released and supplement the numbers in the wild. The second point would be it enables research on basic biology of species. Many species are elusive and have not previously been kept in captivity. So we can know a lot of their biology and their conservation needs once we know what they require. The third would be the captive population can eventually reduce the need to collect individuals from the wild, and it can be sent to zoos and other parks instead of capturing new individuals from the wild. And fourth would be the captive colonies can be used to educate public about the species, its habitat, and to build support for its conservation. There are different problems in managing a conservation breeding program. The number one problem is, where do you get the source stock from? As most of the sources of the populations are so low that if you take them from the wild, the numbers in the wild will reduce drastically. The second problem that they often face is facility that is required, which do the breeding and run the project. That is, zoos, aquaria, and other facilities are required for this program. The third is that we will need a large population size to prevent problems of genetic drift and loss of variability, which is a particular problem with large vertebrates in captivity. And the fourth is the multiple generations will be in captivity and they will start adapting to the captive conditions, leaving them maladapted to surviving in the wild. There's also a loss of learned behavior that can occur in unnatural conditions for them. And they are also susceptible to diseases that are because they are kept in a highly dense environment, high concentration of individuals in a smaller area compared to what they would be in the wild, where one individual does not come in contact with the other individual. But since the area will not be huge, they will be interacting with each other more frequently, and there will be a chance of high disease spread from these individuals. And also some species are very difficult to breed under captive conditions. They require environment which will stimulate them to breed. Like some species require high rainfall, or they require the stimulus of other breeding individuals for them to come into the breeding plumage and start breeding. So these are the major problems that come with conservation breeding programs. So when you start a conservation program, what are the decisions that you need to take before you start the program? Where the source founders come from. So it is important to source the individuals from a population which is large in number and which is diverse in genetic variability. So it is important to source and capture the genes of the whole species. So it can be from various different populations without hampering the major population in the wild. It is also important to know where you want to keep the conservation breeding facility, where do you want to locate it. For that, there are multiple criteria that are involved. That is, should it be in the range of the species or should it be away from the range of the species? It is easier always to have a conservation breeding program in the range of the species. Like if there is a native Indian species, it is better to manage it in the Indian subcontinent rather than taking it outside. It is also important to know the proximity of the source population. So if there is a population like in a region in Rajasthan, it is better to keep it close to the source population. Also, the third important part is where is the land available for constructing such a facility. It is not easy to get land in this day and age, and wherever there is you have to see the pros and cons for selecting the land that is available. And it should also be accessible. That means that there should be connectivity through road, rail, or any other means to reach that area. So how will you transport the source individuals, or how will you transport them back into the wild? All that accessibility needs to be considered from the conservation breeding facility. Along with the logistics like electricity, water, and fuel should be readily available and are essential for running a successful breeding program. Along with that, it is important to insulate the population from diseases, predators, and disturbances. So it is important to manage the facility such that there is minimum disease spread and predators that will come and hamper the population that is kept in the wild. And it is important to have the community support. It is very important to have the community that you're working with support the project. So there needs to be a positive community support in that area. And local market is also very important to get small things. It is important that there is a place nearby and you don't have to go miles before you reach a place to get the basic amenities. So the third thing is making the budget. So for that, you need the capital that will be for the infrastructure and equipment. That is the major costing of the project will be on the infrastructure and the equipment that will be required. Then there will be a recurring cost, that is the staff salaries that will go, and facility, that maintenance of the facility, and the supplies that are required daily, like the food, accessories, and sanitation. Along with that, it is important to research, train, and document the stages of the project. So there needs to be a fund allocated for that purposes. So after the budget is decided, and it is important to figure out which multiple components will be involved and who are the stakeholders of the project. So there will be a MOU which will be signed, which is generally in the case of India, it is with the forest departments of the range states, the central government, and the implementing agencies. So the MOU needs to be finalized, and after that, for working in wildlife, there are permits that will be required from the Chief Wildlife Warden and the Central Ministry to do any project related to wildlife. After that, the land can be allocated for the project and the legal processes can continue. After that, it is important to train the staff for capacity building, that is in the existing facilities. If there are existing facilities for the species or in similar species, it is important that the staff goes and trains in those and then implements the project with whatever modified changes that are required. And they gain as much knowledge related to the species. It can be from the wild, like what is their habitat, what do they actually require, what is the feeding that is required. All that the staff needs to be trained before the project starts. And then there is a learning phase where the pilot implementation is done, and then there is some tweaks that are required in the conservation breeding facility, which will be implemented in the future. It is then comes the management aspect of the conservation breeding programs. There is a very important aspect is the genetic management. The founder population needs to be checked and managed in a genetic way so that they are not inbreeding. There is minimum inbreeding, and the pedigree needs to be analyzed, and as much as possible, the genetic variation is kept in the captive population. The population management, the viability and the demographic projection should be done to see what is the number that you require to be kept in captivity. The other thing is then you have to decide on the housing. The housing is decided based on the balance of all the needs that the species has, along with safety, biosecurity, logistics, and the space that is available. The wild species will be on a large area, but you cannot have that large area in captivity. So you have to compromise certain aspects and make changes according to what would be the best solution for housing those animals or birds in captivity. Nutrition is also an important management aspect. The balanced, as much as balanced diet is required, would be supplemented either with pellets or meat, depending on the species requirement. Breeding is also something that will have to be modified based on how the bird or animal is reacting in captivity. It can be natural. It is best to have natural breeding. If the animals and birds breed by natural means, then it is the best. But if they do not breed naturally because they do not have the stimulus that they have in the wild, there is a high chance that they will require assisted reproduction. The next aspect would be biosecurity. It is important to have minimum contamination in the area that the animals and birds are kept in. We know that they are wild animals and they are supposed to be hardy, but it is best that they are given an environment that causes minimum risk to them. And it is important to predator-proof that area, because one predator coming in would destroy the whole population, and it would give a toss to the conservation breeding program. It is also important to handle these birds regularly and medicate them and transport them. So it is important to train all of these animals to be handled for future. Along with that, there is food production. That is, what can you produce in the local area and can use that. So most of the conservation breeding facilities will have an area where they're growing their own plants, or they're having their own live feed unit, which will supplement the food that is required by the animals. The final stage would be reintroduction. That is, without reintroduction from captivity to wild, the conservation breeding program is a failure. So there is a lot of programs going on conservation breeding, but only 11% have succeeded in establishing a viable population in the wild. Most attempts have failed because the original threat that is causing the species to decline has not been removed from the wild. The composition of individuals that need to be released need to be considered, and a large number of individuals need to be released in a phased manner. It's not a one-time thing that you reintroduce and the population will sustain. It has to be a gradual process, and it has to be released in a phased manner. So before reintroduction, it is important to secure the environment that they will be in. So if they're released in the wild, the threats should be removed from the wild where they are going to be released. It is important to medically screen those animals or birds so that you are not introducing any new diseases into the wild. And it is important to pre and post-release screening is important for knowing what all is happening in the population once they are released. There is a chance that the members that are released will not adapt immediately and might need provisioning of food over a small period of time, and it might be a soft release program. And after the release, it is important to monitor the population and see if they are surviving, if they are able to hunt, if they are able to avoid predation, and monitor if the population that has been released is successful or not. And after that, it is also the part that local human population needs to support the conservation. If they are not there, and how many ever species are released in the wild, if they do not support the conservation, there's a high chance that they will be wiped out again. So these are some global examples of conservation breeding programs that have been successful. So on the top left is the Arabian Oryx reintroduction program, which is one of the first successful breeding programs. This ungulate is from the Arabian Peninsula, and they were in large numbers until 1972. Their numbers declined due to hunting. Then the Phoenix Zoo and many other captive zoo facilities and zoos contributed to their conservation, and from 9 individuals, over 200 young were successfully bred. These oryx were released into the wild again, and since 1982, they were reintroduced in Oman and other areas, and their numbers increased over the next 2 generations in Saudi Arabia, Oman, Israel, and their total population now is approximately 1,100 individuals. The picture on the top right is of the California condor. We'll talk about it a little later. And the bottom left is the black-footed ferret, which is also another example of a species being wiped out from the wild and kept in captivity and bred and released back into the wild. The one in the middle is the Hubara bustard that I earlier talked about. It is a bustard species which has conservation breeding as one of the aspects of preserving, along with preservation of the environment and multiple other facets to it. The one on the bottom right is the red wolf, which is one of the 2 species of wolf in North America, and it is one of the most endangered wild canids. The population decimated in 1960s due to loss of habitat and intensive management of the predators in that area. And since then, the 17 remaining wolves were kept in captivity, bred, and now in the late 2010, 2015, they have started reintroducing them back into the wild, with 1 individual which has been successfully released back into the wild. So this example is of the California condor, which is a New World vulture species. It is found in North America. The species has declined in the last 100 years due to habitat destruction, shooting, and poisoning. There was a recovery team which suggested that there should be a multi-pronged approach to preserving this species. So they recommended habitat studies, protection, along with radiotelemetry and captive breeding program. So by radiotelemetry from the wild, they found out why the population was declining. It was due to lead poisoning of the carcasses that were shot, and hunting is legal in many of the countries, and it is one of the favorite hobbies of many individuals. So they used to use lead for hunting. The gun would be used, and that caused poisoning of the carcasses and declined the numbers drastically. So then the birds were taken into captivity in 1985, and from 9 individuals, the captive stock had increased to 49 condors. And after that, when the release program started, they avoided imprinting by feeding the chicks with glove puppets and cross-fostering by another condor species. So reintroduction began in 1991 with soft release, and the program has been going on, and it's one of the most expensive captive breeding programs, which has costed almost $35 million since 1987. And it is still continuing with the reintroduction, still continuing in the wild. So this is a ferret, that is a black-footed ferret, and it is also a species which was in the US. which is in the US, and it was a widespread species, but the numbers declined in 1960 due to depletion of its prey, that is the prairie dog. As you see, the rodent below in the picture is the prairie dog. The prairie dog's number declined because humans started occupying the lands and using it for agriculture. So they were hunted and removed from the areas that were originally found in. The reduce in prairie dog numbers caused the reduction in the numbers of the black-footed ferrets, so much so that only 10 individuals were left in the wild, and all the 10 individuals were then taken into captivity. And from them, the captive breeding program produced 2,600 kits across 7 institutions till 2002. The reintroduction started in 1998, and 873 ferrets were released back into the wild. But they still face challenges because their populations cannot cope with the loss of habitat that has still continued, and the prey is still reduced in numbers, along with other aspects like diseases that are introduced by the non-native fauna and flora, and the loss of survival skills, because that happens when the animal is kept in captivity for a long period. They do not know how to hunt. They are not afraid of humans, and that causes the less chances of the species to survive in the wild. So the conservation breeding programs in India have started quite recently when you consider it to the other parts of the world, but we are still doing a lot of conservation, and we have a lot of species that require conservation breeding programs. So the picture on the top left is of the Western Tragopan, and it has a breeding center in Sarahan in Himachal Pradesh. It started with a stock of 8 individuals and reached 40 in 2018. The natural populations are very small and highly fragmented, and in the Himachal, because of the fragmented populations, there is reduction in the quality of habitat, and so the species range, which is already a restricted range, it has shrunk even more. So this is established to have a reserve population of the species in captivity. Similarly, the vultures in India have, as I said earlier, the numbers declined due to diclofenac poisoning, and in veterinary use, due to veterinary use of diclofenac, the numbers reduced. And you have a talk, I think, by somebody who will be giving a talk to you on the vulture breeding program in India. So they'll tell you in detail about the breeding project, but the Pinjore Breeding Center in Haryana for vultures is one of the best facilities, which is producing the chicks in the captive program, and they are planning on releasing more and more vultures back into the wild. The one in the bottom left is the lesser known pygmy hog. So this species is found in Assam, and they require, this is a critically endangered species which requires tall, wet grassland areas, and these tall, wet grassland areas have reduced, so their habitat has shrunk, and a captive breeding program was started from a population that was captured from Manas, and since 2001, the total population in captivity has steadily grown to 77. There are many zoos which are now contributing to conservation, where they're starting a conservation breeding program, like the Darjeeling Zoo, which has a conservation breeding program for the snow leopard and the red panda. So now I will talk about the bustard recovery program that I am working on. It has started since the past 2 years. The conservation breeding aspect of the program has started since the past 2 years. It is a multi-institutional initiative that involves the Ministry of Environment, Forest and Climate Change, Rajasthan Forest Department, and the implementing agency is the Wildlife Institute of India, along with collaboration from International Fund for Hubara Conservation, which is a conservation breeding facility in Abu Dhabi for the Hubara bustard. So since they are bustard species, they are similar, and we get to learn a lot from them, from their experiences, and we modify and use it according to what is required here. So what are bustards? Bustards are huge omnivorous terrestrial birds of open country. That is, they were found in the desert and grassland areas. They have 3 toes, but they do not have an opposable thumb. That is, they are meant for just staying on the ground. They do not have any grasping aspect. So they are mostly found on the ground and they take short flies whenever possible. They are heavy birds, Great Indian bustards, so their weight varies from 4 to 10, 15 kgs. The males can grow up to 12, 15 kg weight. So they are huge birds and they have one of the most astonishing displays during the breeding season. As you see the picture on the right is of a male Great Indian bustard in the display plumage. It has a huge gular pouch, which is inflated by air, and then it makes a booming sound to attract the females. The species was once thought that it needed protection, and so Salim Ali and other ornithologists asked that it be the national bird, but unfortunately, the peacock was selected as the national bird. But this bird has been given special importance in Rajasthan, and it is the state bird of Rajasthan. So why the numbers are, so it's important to know why the numbers are reducing. And we all know that all the animals, the numbers are reducing, but how much has been the reduction in the range and in the population? It is important to know that. So since 1969, the numbers have reduced, and since the last survey that was done in 2017, an estimated population of 140 individuals remain in the wild. So the total population of Great Indian bustard in the wild all over the world is reduced to 140 individuals, and majority of it, that is approximately 100, is found in Rajasthan, with a few numbers that is in Gujarat, in the other range states that were earlier also having grassland areas, that is Rajasthan, Maharashtra, Madhya Pradesh, Karnataka, and Andhra, have small populations of a few individuals, but unfortunately there are no males, or their females are laying infertile eggs in all the other populations. So the majority of the population is in Rajasthan, and hence the program was selected to be started in Rajasthan with the major population stronghold there. So why has the population crashed in the past years? In the past few years, it is mainly because the grasslands were not preserved and not given the importance the forests were given, because grasslands were considered wastelands and they were given up, and either they were planted with trees or they were given for another aspect is the green energy which they were given to. So a lot of windmills have come up, a lot of solar projects are coming up in the grassland and desert areas, which is the stronghold for the Great Indian bustard right now. They are a case selected species, which means they have a poor recruitment. They lay one egg in a year and one chick will survive with one breeding season, successful breeding season. And if the chick survives for approximately 250, 300 days, there's a high chance that it will survive for the rest of its life for the adult age. But they have a poor recruitment, but once they are recruited, there is a high chances of survival. But this species has adapted in so many years that it is a grassland species and does not have any barriers in the sky. It has never evolved to have barriers in the sky, but with the power lines coming up, they have a very poor frontal vision, so they don't see the power lines and they hit and collide with the power lines, and that is causing a major threat to this species right now in the conservation aspect. With approximately 2 deaths that we have found every year due to power line collision in these bustards, which already number so few. Along with that, there is a compounded effect of hunting and habitat loss on the slow life history. So there is hunting which is going on, illegal hunting, which we can try and stop, and there is a population which is said to used to exist in Pakistan, but now it is so porous that there are no borders for birds, so they can fly and go into Pakistan, and if they're shot there also it is illegal to hunt it, but there is a chance that it will be hunted. Along with that, the intensive management of agriculture, that is a lot of pesticides are being used. So it is an omnivorous bird which feeds on insects and requires high protein diet, especially in the younger age. So with a lot of pesticides and insecticides used in farming, it reduces the prey for the bustard and causes loss in the numbers because of that as well. Along with that, the few grasslands that are left in the habitat are overgrazed by the amount of livestock pressure that is there in those areas. And so they have a multiple threat scenario for them ahead of them. Apart from, like, let's consider this graph, which shows the abundance and the years. So if there is 2 human-induced deaths per year, the population of approximately 140 right now will reduce to 50 by the next 50 years, 50 to 20 individuals in the next 50 years. But even if there is no human-induced death, which is very unlikely, and even without that, the population will not increase rapidly because it is a very slow growing species, and hence it is important to have a captive breeding program to supplement the numbers even along with protection of the habitat. So what do we do in the conservation breeding program is we need to find a founder population. To have any wild animal or bird which has grown in the wild is very difficult because they do not breed in captivity and they are very skittish and get injured very easily when kept in captivity. So it is important to have either chicks or eggs from the wild and rear them so that they are used to the captive environment and they will breed. So since 2019, June was the time that we started collecting eggs. And we right now currently have got 16 individuals in captivity. So the field biologists will go and look for nesting females. They have a particular behavior and they do not have a nest per se. So they lay the egg in the open in a area and then they incubate that. So they roam around in a peripheral area. And so you locate the female and then try and reach the spot where female is mostly found, and you collect the eggs from that area. Then we put the egg in a cool box with, so a cool box is basically an insulated box with sponge so that there is minimum jerk and there is no chance, there should be reduction in any chances of mortality while carrying the egg, as any jerks would cause mortality. So they are brought in a very slow moving vehicle at 5 kilometers per hour. The vehicle moves for approximately 40 kilometers and brings it to the conservation breeding facility. So the temporary facility has been established at Sum in the Desert National Park in Jaisalmer. So these, as you see, are the tourist huts that the forest department had, and these were the guard chowkis. So they were converted into temporary incubation and rearing units. This is the juvenile unit, and these are the tunnels that are set for the adult birds to stay. This is the quarantine unit with the live feed production and an exercise cage for them. So this was set up within a few months and we need it immediately so that we don't lose the next few years till the facility is built. So we built a temporary facility in Sam, and there is a main facility being constructed in Ramdevra. So once the egg is brought in, they are kept in incubators and with temperature and humidity control. They are kept at 37.5 degree temperature, and the daily density loss is recorded by measuring the weights, candling, and it is important to rotate the eggs. As they are very heavy eggs, there is a chance that the embryo will stick on one side. So it is important to rotate the eggs. The incubators do the manual rotation, but in case they don't, we rotate the eggs whenever we take it out of the incubator to do the measurements. Egg temperature is also recorded, and as the days progress, the temperature as the embryo develops, the temperature increases slightly. And just before pipping, there is some movement in the egg and there is vocalization in the egg once the internal pip has occurred. So after the pip has occurred, we move them to the hatcher, which has a slightly higher humidity. And the estimated initial egg mass is 135 grams. And by the time the chick is hatched, it is approximately 89 grams in weight. So it loses that much moisture over the period of 21 days. And the internal pip is generally at 21 days, and by 22 days, the chick has hatched. The chick is then kept in the brooder, which is again a temperature and humidity controlled environment, because it is important for birds to be kept in a temperature and humidity controlled environment, as they have a higher body temperature, and the mother would generally keep it under the feathers and keep it warm. Then after the first day is over, they're not fed anything in the first day, as they already have yolk sac, which will provide for them for a day till that is absorbed. we do not provide any food. After that, we give after the first day, the second day, we provide food gradually, increasing the quantity and reducing the number of meals. In the first stage, they are kept in an indoor environment in these chick boxes. As you see, they're transparent boxes so that they can see each other and they can see the keepers that are handling them. They also have a temperature heat lamp that will give the bird enough heat. And if they want to move away from the heat, there is enough space for them to move around. And it is important to imprint them at this stage, as imprinting of the founder population is very important. Otherwise, they become scared of humans and there is a chance that they will not allow handling, which is very important for the founder population. After that, they are moved into the second age, which is a temporary temperature moderated environment, which is outdoors, but with shade on the top, so that they have some areas so they are kept in smaller cages with one-to-one keeper interaction, and the imprinting continues, as this species is a species that stays with the mother for more than a year. The chick will follow and eat what the mother is eating, and the mother feeds the chick or shows what is to be eaten and what is to be done. So that's why their imprinting process is a long-term process, but it will vary according to the species that is kept in captivity. So after day 60, they are moved to these tunnels with lesser interaction. It's not completely off. The interaction is less than what it was earlier with the keepers, but they will interact with the keepers and with other birds. And as you see, there are, this is a female bustard, while this is a male bustard. They're almost double the size in weight. So conservation breeding, there is a important aspect is feeding and the growth of the bustards in the initial period. They need to be fed a nutritionally balanced diet. It is important to do that. And since they are omnivorous birds, they are fed on omnivorous diet pellets that is obtained from the US. It's a Missouri product, and it is made of invertebrates and fiber. When they are young, they are fed 9 meals in a day, and as gradually as they grow older, they require lesser meals as they eat more in quantity. And the feeding will depend on the species that is kept in captivity. And for these birds, they are mostly active during the morning period and during late evening. So those are the peak times when they will eat more. And in between, you can feed them something that they like eating, so it's easier to feed, like mealworms or crickets are something that they love to eat, so that they'll eat even during the hottest period of the day, but they will not eat the nutritionally balanced food during the hot period of the day. And it is important to hand feed them and stimulate them and transition them into the adult diet gradually. So the daily weight gain is like a linear phase for them to grow. The females grow like this for approximately 140 to 160 days. And the male keeps growing till it reaches adult weight, approximately 10 to 12 kgs. It has reached after a year. So till the 1 year, their growth period is like a straight incline. So we do not have a record of Great Indian bustards in captivity, so their growth record in captivity. So we compare it with other bustard species and see if their growth rate is normal. So it is the Great Indian bustard, that is Adiotis nitriceps. The growth is very similar to the Kori bustard, which is also a large bustard species. And they have grown both linearly, along with Arabian bustard, which stagnates at approximately 28, 35 days. The growth reduces, while in Kori bustard and Great Indian bustard, it keeps increasing. So they are fed with forceps, and they are shown like forceps are similar to the beaks of the mother, and that will show them what is the food. You point the food and then you put it on the ground and let the chick eat by itself. So this is generally the plate that is offered. This is the omnivorous pellet that is soaked in water and it becomes soft. This is the hard pellet that is given to the adult birds, and the birds are slowly transitioned into this pellet, along with mealworms and superworms that are being grown in the facility. And alfalfa is the green alfalfa and mustard they love. So those are grown in the facility and without any pesticide and are fed to the birds. So other husbandry aspects include a strong disinfection protocol. The emphasis on disinfection is maximum, as even a smallest mistake will cause the population in captivity to be wiped out, and it will not be possible to regain the population if there is a breach of contamination, or if there is a chance that there is a outbreak which will occur due to carelessness of disinfection protocol. There is also a hygienic live food production, which requires a separate unit of staff which will work in the live feed. So we have a mealworm room and a cricket room and a farm that grows mustard, alfalfa, and the seasonal fruit that are required daily. Along with that, it is important to get the birds to exercise, because the chicks will start following the mother at an early age and they will start moving. So for their development, it is important that they move around so that the skeleton and the muscles develop accordingly. We do the vaccination of birds here for ranikhet and pox. That is because there is no major poultry industry around here. So the chances of the diseases transmitting from poultry are very less. But as precaution, we take the vaccine of pox and ranikhet here, because in the wild we have seen a few sparrows with pox lesions. So whatever is the environment that you're keeping them in, what all diseases are prevalent in those environments, it is important to vaccinate the birds or animals according to that. And there is also regular wing trimming done to avoid collision induced trauma. So if they take major big flights, there is a chance that they will fall harder and cause injuries. So the wing trimming is done on a monthly basis, so that the wings are kept, the feathers are trimmed so that they do not take big flights. They are able to take smaller flights, but not big enough flights to get injured. And there is importance of recording all the data and keeping all the data, that is the morphometry, weights, and the food consumption, to know what is the growth rate, and if a bird is not doing well, to compare it with other birds in captivity and to have a baseline data for future. So for us now, the way ahead is to have a population which will breed. These birds do not start breeding before 4 years. So we have to wait 4 years for them to start breeding. And we have intensive individual management. Each individual is given sorry, did I overshoot the time?
Hello, ma'am. Are you able to hear us? Hello.
Yes, I can hear you. Can I start? Sorry?
You're left with 5 more minutes, ma'am.
Yeah, sure. I'll just finish off with a small video on it, and then that will be the end of the presentation.
Okay.
So I would like to share the video of the conservation breeding program that is currently being run in Jaisalmer. It's a short 5-minute video, so just go ahead and See.
Mustards are one of the heaviest flying work. Mustards are one of the heaviest flying works. They have no frontal vision, so that they fly with a long expect and one problem causes most of their deaths. As you can see, mustard there was flying all sharp source of heat. Considering the declining population of the mustard, the issue of wildlife was managed and it benefits of conservation and insurance and needs to extinction. We will take you through the conservation training facility set up at some to represent natural park, collaboration with the government of Rajasthan with the wildlife insurance and temporary facility set up at some. Including government portals and some were transformed into conservation training center. With the help of the international farm scientists from NAHEP-IDP helped us in designing and setting up some. The facility here includes subsequently there is a continuing use the communication center and the field facility is also created. Another kind of access the eggs or the chicks and we can rear them with proteins. The process commences with the location of the nesting. And this required a lot of from very good. We had trained experts from IFXC assisted our team in locating the nest. They did the room and centric living place allows them to so our team was able to locate several nests in China. Once the egg is collected, it is in and transported from the nest site to the center terminal. common population or the
Okay. So I think I can just say, I'm sorry, I got to know that the video is not working well. So you can see the video of the Bustard Recovery Program on YouTube. It is there. The Conservation Reading Center for Great Indian Bustard at some Jaisalmer. So this is the team that works in the field and on the Conservation Reading Program. Thank you for listening. And you can email me if there is any queries or if you have any questions, we can have a session right now.
Thank you, Dr. Krishna Madam. You have nicely presented regarding this bustard project. First of all, our respected principal and dean, Dr. P.S. Sir, P. Staff Sir, has joined this training program. So I welcome him. I also welcome Dr. Odedra Sir.
Sorry.
And co-chair Dr. Kepa Sir. So, dear students, trainees and friends, if you have any question regarding this training lecture, you are most welcome.
Hello, Dr. Krishna
Karkaria, right.
Right in
It's Karkaria, ji sir.
Madam, you have been given an information since our joining the science that principle of evolution and the survival of fittest over a period of many centuries and beyond.
Right.
The creatures, especially the living creatures, may it be plant, animal, birds, trees, human, they have undergone various challenges that, and once upon a time they become under endangered or extinction. And some survive, some decrease. And if the aided breeding in captivity or conservation and all may reduce the decreasing numbers, or instead you can make them to be remain present on the earth. But will you be in position to make them to be fitted for future propagation naturally?
Okay. So basically a question, sir, is that if the captive bred population will survive in the wild, and if it is a good idea to keep a captive population for conservation, as they take a long time to adapt and evolve. I agree completely, sir, with you that evolution takes generations and generations, and the changes in the environment are very gradual, but with the current scenario, the changes are so rapid that the most of the animals and birds will not be able to survive the changes that are happening due to the anthropogenic pressures that are there currently, the animals and birds are facing currently. So the conservation breeding program is basically to have a captive population and to reintroduce them in the wild. But there is a chance that the captive population might not breed successfully in the wild. But the chance has to be taken at this stage, because if we don't take any, there is a very high chance that they will be extinct in the wild. So whatever measures can be taken at the current stage and what techniques can be employed to breed them, it can be for the future to learn as to what is the best method for them. And it's very important to preserve the wild habitat, as they will not breed if the wild habitat is not secure, and they will not breed successfully if there are very few individuals that they do not have a mate choice. Then there is a chance that the population in the wild will not be breeding. Right, sir.
And at what number of population or the stage the animal or bird is classified as endangered or critically endangered, like that? Are there any specific scientific information
So for
that
each,
beyond? Yeah,
so
yeah, please.
IUCN, the red list is categorized based on the numbers, but the numbers is not a fixed number. Like for captive breeding, it is, like I said in the, if it is less than 1,000 individuals, they should be considered for captive breeding. But making them endangered or critically endangered is based on the species. They, for some species, they are less than 400, and so they are called critically endangered. For some species which are already occupying a very specific habitat, they might not be considered endangered if they are less than 1,000 individuals also. So the IUCN, the red list is, they categorize based on each species in the environment, and it is based on the where it is found. So they might be extinct in certain parts of India, but they are still present in certain parts of India. So they are not extinct in the wild, but they're extinct in certain regions in the wild. So they'll be classified based on the region as well as the extent of the species.
What is your speculation about the survival of this bustard, which is almost around 140-50, as you narrated, in the natural?
So we are positive and hence are working on the project, and we are trying to mitigate the threats in the wild as well. We are trying to get the central government to put diverters on the power lines which are causing a major threat for the bus, along with certain areas which are being fenced, and grasslands are being cultivated in those areas, so the birds can use those areas more frequently. So we have a multi-pronged approach. Conservation breeding is one of the aspects, along with preserving the environment for the future release of the birds. So we are hopeful that with the population that we have, we will try and increase the numbers and hopefully protect the environment long enough for the future generations to be released in the wild.
Okay, this census was conducted by physical observation or some geospatial?
So the population numbers was done by doing a line transect survey. So the habitat of the Great Indian Bustard was covered by line transects, and the numbers, individuals were counted. So the approximate numbers of the individuals are 140, but in Rajasthan it is estimated in two areas, that is the field firing range and the desert national parks holds the major population of approximately 40 individuals in each of these ranges. But in many of the other ranges, like in Gujarat and all, the population has declined drastically, and now there is no male in the Gujarat population in Kutch, which is a very sad situation for the birds, as they are laying infertile eggs, but there is no male to help them grow their population.
Single population in the Kutch near the South Rajasthan border.
Right, sir.
And also wants that this to survive. And we appreciate your center's efforts for putting a lot of plans and projects and activities to survive this endangered species. You should take an example that in each forest also once upon a time, ascetic land depleted to around 50 only, being higher hierarchy in the vertebrate animals. Now it has close more than 600. So, with the scientific efforts and social affections and governments and the veterinarians and all together, this species will also revive.
Yes, sir. We are all hopeful that it's inspirational. Other species survive and are brought back from very few individuals. It is great to see that. We are hoping the same for the Great Indian Bustard as well. Thank you, sir.
Are you any sub-centers at Gujarat?
No, sir, we do not have any center in Gujarat yet. We were working on a project for the lesser Florican breeding program. but I think it's started separately by the Gujarat Forest Department.
Why don't you involve veterinary college? Then preference should be given to those who are much technically involved in breeding and policies and reproduction and management and all.
Yes, sir. It is important. Veterinary is a very important aspect in conservation breeding. But along with that, there are multiple agencies that are involved, and field conservation biologists as well are very important for the conservation breeding projects, as they are the ones which will secure the population. And it's important that the support is generally lacking from the agencies when it comes to contributing. So if like we would love to work with veterinary universities, especially for screening of diseases, and if there is any help that can be provided, that would be great.
In case in future, if you find some prospects where the veterinary college expertise can be put to a partnership, either as sub-center or as associate workers or whatever way.
Would be great, sir.
So now 4 veterinary colleges in
Yes, sir.
Gujarat and one Nagar near Dantewada, which is at the border of Mount Abu, is very near to Rajasthan. And one more is coming next year at Junagadh, which is also joining the border of Rajasthan. But Kutch, where these birds are found, have more geographical and cultural relation with the Saurashtra. And Junagadh is open for you for either the health management or breeding and all. So remember us.
Thank you so much, sir. Yes, sir. We'll definitely collaborate for future.
Thank you, madam.
Thank you, sir.
Madam, I have one question.
Yes, sir.
As you have said that there is no male population of Kutch. So, cannot you translocate few males in that population for their assisting in the reproduction?
So that is one thing that can be done for conservation, but since the population is so low and the friction between the Rajasthan and the Gujarat government is so, as we've seen earlier, like how the lion is not being pulled out of Gujarat, it is the same situation in Rajasthan. If there is no government agencies are not willing to cooperate with each other to get the populations of the wild animals, and the wild animals are suffering because of that. It is a very sad situation. We would like that at least if not the females brought to Rajasthan or the male being transported to Gujarat, that would be ideally the best scenario.
That is a very pitiable situation, madam.
These birds are also in the Sindh area of the Pakistan.
Yes, sir. There are. Yes, sir. Some reports are there of hunting as well, but they are not monitored regularly. So there is a chance that the population from Rajasthan and Gujarat is going to the to certain grasslands in Pakistan, the Cholistan grassland.
When birds are going to Pakistan, ask them to come to Gujarat also. Let government see it. They do not require visa and permission.
Sure, so we'll train them to go to Rajasthan if it is possible.
Hello, ma'am. I have also one question. Hello.
Yes, please go ahead.
Yes, ma'am. I want to ask that you have told that these captive breeding programs are majorly started with very few individuals, just like in 10 or 20 like, and reaching up to 100 of the individuals after the breeding to release them in wild. So what about the ensuring genetic variability, because producing the 1,000 animal or 100 animal from these tens of the population? So what about this?
It is a major factor. So the founder population, which is already, it's going to be a small number because we can't have the majority of the population from the wild. Many breeding programs started with just 10-12 individuals that were remaining. So the population was wiped clean and they had no option. So they had only 12 individuals to start with. So for that, we can't help with the less genetic variation present in that population. But for breeding programs like the mustard breeding program, it is important that the genetic diversity of the population that is currently present is taken in account. That is, you don't take eggs or chicks from the same mother in the multiple years that you're collecting the founder population. It is important that you select different individuals and, if possible, from different regions, so that you cover a large area and select the maximum genetic in the wild for your founder population, so that there is and keep a number. And then there is also a requirement to supplement from the wild. Even after a few years, if you can supplement a few individuals from the wild and use those, it is important to reduce the genetic depression.
One more last question, madam. Are there any efforts or plan for repository and depository of the germplasm of this bird somewhere in the in vitro situation, so that it was
So the gene preservation for the bustards, we are collecting as much material from the wild, like the feathers and the fecal samples, and seeing and keeping there is a genetic depository at Wildlife Institute of India, and they're taking we're keeping as many samples as possible from the captive and the wild population, so that we have the capture of the genetic diversity. But we have not collected sperms or ova from the birds, as it has not occurred till now. We've just found dead birds from the wild, and we have collected eggs. So we haven't got the opportunity to go further and dive into the artificial preservation of the germplasm.
when males are less in number, and you have the male also somewhere in Rajasthan in nature and in captivity, are there any possibility of collecting sperms and then freezing it and then to use it somewhere in captivity where the male population is depleting or no presence?
So it is very important question that male germplasm be preserved, because males are the most affected of the species. Like majority of the male deaths are in the power lines compared to the females. So the availability of sperm will be a very important factor that will play in future breeding. So we do want to preserve so the captive birds, they have not matured yet. They take 4 years to mature, and they have a short breeding season where they will be displaying. And after like in wild, at least we have not seen in captivity. So it's a learning process, and we'll see how it goes with each coming year. But in the wild, the males start displaying at 4 years, and the biggest, strongest male is generally 5 to 7 years old, which will be breeding with majority of the females in that area. So they have a exploded leg system, and if we go near a breeding male, it will immediately go away from the breeding, from the display area. So it is important. We don't know how to protect without the hampering the breeding population in the wild. But if we can work on a way that we can collect semen from the wild birds for future, it would be great. But it is a give and take. And right now, to secure, even right now, if we have a population which is starting to breed, we can think about doing that. But right now, the females are not mature in captivity. So it will take a while for them to mature. And after that, we'll start with the breeding efforts. But yes, sir, it's if there is a way to preserve the wild spermatozoa of male, would be great.
And your center must also have some connectivity with philanthropies and NGOs, those who are working for the bird conservation or supporting on the philanthropic platform. Are there any MOUs or connections or partnership or putting social efforts in addition to this scientific efforts in captivity?
Our project does want to do a lot in the social aspect, but with the minimum staff that we have, it is not possible for us to do a lot with the local community. But we have outreach who goes and figures out what is needed by the villages surrounding those areas, and they then communicate to other agencies or other NGOs what the community requires, and if possible to connect the NGOs with the local community. But we are not directly associated with the community aspect.
And one more, last.
Yes, sir.
We under Veterinary Council of India have got 1 year internship in last year for graduating students.
Yes, sir.
And now it is mandatory to send our.
I'm sorry, I can't hear. It has stopped for a second. Can you hear me? I think it has freezed.
ત્યાં ચાલુ હશે ને એટલે અહીંયા બેન્ડ ઓછા કર્યા છે.
નહીં સર લાઈટ ઇન્ટરપ્શન એટલું કન્ઝપ્શન
Hello. Hello, Krishna. Am I audible there? Left.
So, dear students, trainees and friends, I am really thankful to Dr. Tushna. She has focused the economics of conservation program, project management. Then she has focused on particularly great Indian bustards, and she had narrated various breeding practices. So I am really thankful on behalf of these organizers. Now, friends, we have a second lecture on captive breeding and reintroduction of endangered wildlife. So, so we have a very eminent speaker and very respected speaker, Dr. P.H. Vatalia sir. He is working as a Director Extension Education and ex-Vice Chancellor of this Kamdenu University. So I welcome you, sir. Dr. Vatalia sir.
Yeah, thank you. Good morning to all.
So, yes, sir. Thank you, sir. So, dear friends, today we are having a very respected speaker with us. So, I invite him, and first of all, before invitation for a lecture, I invite my colleague, Dr. Shephar, to introduce Dr. P.H. Vatalia.
Good morning, sir.
Good morning.
Thank you, sir. Okay. So before starting the lecture, first of all, I will give the brief introduction of the Dr. Vatalia sir. So Vatalia sir has completed his BVSc and NAHEP from the College of Veterinary Science and Animal Husbandry from Gujarat Agriculture University in the 1983. After that, he has completed his master degree in animal genetics and breeding in 1987. And subsequently, again, he has completed his PhD from the same university in the year of 1994. Vatalia sir is very renowned personality and is a vast experience in the animal genetics fraternity. And he has worked in the different capacity, right from the veterinary officer to ex-vice chancellor in the Kamden University, and right at present he is working as a director of extension education. During his career, he has published the 44. He has completed 44 research projects, and he is given the characterization for the very important breeds of the Gujarat, especially gir cattle, buffalo, sheep, and goat. He has also conducted the several national and international trainings, seminar, symposium, and also published the number of papers. And he has also guided the 18 MBSc student and one PhD student. So with this brief introduction, now I request Vatalia sir to give the lectures on the captive breeding and the reintroduction of the endangered wildlife. Sir, please.
Very good morning to all. Am I audible to everybody? Somebody
Yes,
may
sir,
respond.
yes,
Am I audible to everybody?
Yes.
Yeah. If it is okay with the system, then I will start my talk. At the beginning, I thank the organizer, especially the Dean of the Faculty of Veterinary Science at Junagadh Agricultural University, Dr. B.S. Tang, and the organizing secretary, Dr. Anshu Elawat, and the entire team, which has actually thought of having such kind of training program in this time when people don't move from one place to another for any kind of training program. So that too a 5-day vocational training program online on captive breeding and health management of wild animals. So I congratulate the team at Junagadh Veterinary College led by the able leadership of Dr. Tang and well thought of plan of the training program devised by the team led by Dr. Anshu Helawat, Dr. Odedra, and others in the Department of Animal Genetics and Breeding and the entire College of Veterinary Science with the help of the infrastructure that has been made available to you by the university. So I thank you once again to speak to you, the faculty with which I have worked earlier and the students who are at my heart from the beginning of my career. So thank you very much. To start with the topic, I was very much enthused to listen to my previous speaker, Ms. Karkaria. Although I joined in the later part of her presentation, but she had the practical knowledge of conservation because she comes from the Wildlife Institute of India and she is actively engaged in the conservation plan of some of the species that she narrated well. So I am very much delighted to be part of this gathering. To start with, I came across one publication by the Government of India, the Department of Information. And in this month's issue of that publication, New India Samachar, they have 1.5 page devoted to conservation of wildlife, entitled Protecting Wild, Protecting India. The title itself tells that India gives great importance to protection of wildlife and it equates that with protection of India as a country, India in its uniqueness. So this is the importance that is being given to wildlife in this country. It has been mentioned there that the leopard population, the lion population, the unicorn, rhino population, and tiger populations, they are on the rise because of the concerted efforts of various agencies involved in conservation and uh, improvement of the habitat of the wild animals in the country. So this is a very positive sign that is coming up in the national scenario. And for that, we need to congratulate and applaud all who are involved in the process of conservation of the wild in the country, be it they are the breeders, they are the managers, they are the philanthropists or the social workers or the people in the administrative component of the government which provides the logistic support to the efforts of conservation biologists in achieving the target of maintaining the sizable populations of very important species of wildlife which are important as they represent our lifestyle, they represent our culture, they represent our history, and also make India a distinct country that is viewed globally as a unique country in the entire world. I think as per the latest statistics that has been presented to me, I say that the population of leopards has risen over the years, and as of December 2020, the number has risen to 12,852. These are likely to be the estimates. So leopard population has crossed around 12,852. This is a good sign. There are many issues of the leopard hunting within the human habitats, but keeping aside certain stray incidences, in majority of the cases, the threats to the human population are minimized by the respective authorities who control the behavior of the leopards in the wild. Similarly, there is a very rosy picture as regards to the Asiatic lion population. We are proud that lions are found only in two regions of the world. One is the African part, that is savannas, where the African lion is habitated, and second is the proud location of Gujarat, that is in the Gir forest, Gir Abhyaranya, where we find Asiatic lion. As you know, the history of Asiatic lion, over the decades, the population is on the rise. But historically, the population had dwindled or had reduced in the past century to about 20 animals only. That was around 1920, the time when the cattle breeding farm of Junagadh was established. So before 100 years, the recorded population of Asiatic lion was around 20 only. Now we have risen, as per the last census data, we have risen to 674 Asiatic lion as per the last count of the animals conducted in 2020. So it's a tremendous increase over the century. Especially in the last couple of years, the number has risen handsomely. In the last decade itself, it has risen almost doubled. It was around 300 to 325 in 2010, and now it is 674. So this is the sign which indicates that we have made concerted efforts, effective efforts, and result-oriented efforts in conserving Asiatic lion. And now people are starting to talk about the overpopulated habitat of Gir forest as regards to Asiatic lion, and people are asking for translocation of Asiatic lion to other regions of the country, also because of the surplus number of animals which are now moving out of the natural habitat into the human population, especially in the peripheral area of the sanctuary. And sometimes the lions are also found in Jodhpur, Setrunji, and nearby areas also. So this is also a healthy sign. Similarly, tiger population has risen over the years by 33%. So this is rise has been observed in West Bengal. The rise has been observed in Karnataka and also in Madhya Pradesh. So these are the three areas where the tiger population has shown a tremendous increase. About 33% rise in the tiger counts has occurred. Similarly, other wild species are under close control, and they are under the controlled monitoring, so that the populations are not endangered. Similarly, the story about the elephants in the northeastern part as well as in the southern part, the elephant population is being conserved and the population is not under danger. Unicorn, a unique animal that is present in India, that is the rhinos. In the Kaziranga sanctuary, the population has been well conserved. One rhinoceros have reached, actually it reached the lowest population size of around less than 200 animals at the turn of the 20th century. And now there is rise of about 75% in the population in the country. So this is also a healthy sign. Why I am narrating all these things is to put a picture that everything is not bad as regards to the conservation efforts of the conservation of wildlife in India. No doubt, the conservation efforts are time consuming and they are also heavy on resource requirements. Resource in terms of money, resource in terms of the experience, the manpower, as well as resource in terms of the predators for conservation of this particular species concerned in the natural habitat. Also, there are issues related with the human-animal conflict as regards to the wild animals, which sometimes move out of their territory in search of the food animals, and sometimes when not getting the food animals of their choice in the habitat, they sometimes attack the human population, creating a great public distress and sometimes creating doubts into the minds of the general public why we should spend too much for protecting a species which is danger to the life of the people itself. So we have to balance between the need of the society and need of the nature. As you know, of late, we have been talking too much about the negative impact of climate change. None can be said to be free from the impact of climate change. The rains have changed its pattern. The cold waves have, or the cold has changed its pattern. The heat and the level at which the globe is getting warmed has also changed the pattern, and we find mixed seasons in unexpected months of the year. It impacts the flora and fauna of any given geographical area, whether it is India or Africa or Europe or America or Antarctica or the Arctic region. Everywhere the impact of climate is visible. And recent examples of many disasters happening in some parts of the world are indications. To cite the example, the glacier that exploded in the Himachal Pradesh is one example. Similarly, heavy storms happening in the Texas region of United States of America, heavy snowfalls occurring in the areas where there used to be no snowfalls. That is also the situation. Why I'm talking about the climate change is because for survival of any species, whether it is human or wild animals, wild birds, or wild amphibians, or the marine species, natural habitat is important. The sustainable environment is very much important for survival of a species in an environment. Each species is adapted to certain range of climatic parameters which make it thrivable, sustainable, livable. Because survival of any species depends on availability of right environment, right climate, right quality of feed resources or food resources, or what we call as the predator population for the wild, availability, the abundance of the food or feeding material, and the variation in the availability of these resources around the year. It is not only the availability of the resources in a given season important, but it is also required to be available all through the year. That's why when the feed resources are limited in a certain area, the wild populations have the tendency to migrate from the one region to another region. That's why we find the wild birds moving from Siberia to Gujarat in the little run of Kutch or Nal Sarovar area, or the Kutch area, where they come, they migrate to the Gujarat region. They also migrate to Ranthambore area in Rajasthan, and they breed there. When their siblings or their uh, chicks are ready to fly, they fly back to uh, their native. So there are two natives for such kind of migratory birds, and that's how the species survive across the regions of the globe, moving from the temperate region to the tropical region, and then from tropical region to temperate region. So this is the natural way of conservation of the species. When such factors which control or facilitate the survival of the species are affected by one or the other external factor, it creates the disturbances. If there is an ocean which is very much silent, if a heavy storm comes, the waters rise and creates difficulty for the boat that is floating in the ocean, and it is a possibility that it may overturn and the people sailing in the boat may fall and may die. This also is likely to happen to any of the species. A single negative incidence that happens in a particular habitat is likely to create a very big uh, issue for that particular species to survive. I will cite an example of the kangaroos being displaced and many vanishing in Australia because of the wildfire that occurred in Australia last year. And because of the wildfires, many other than the other species other than the kangaroos might have vanished. We do not know, because we might not have identified the species living in the wild, and they might have vanished. So natural disasters are one reasons why the species dwindle in the population. Another reason is the increased human activity in the in the natural habitat area of the wild. Human activity, as you know, globe is one and the land of the globe is not expanding. On the contrary, the seas are rising, the oceans are occupying more and more space in the peripheral area because of the rise of the global temperature, and waters are occupying more space on the earth than the human claims the land for its purpose. So land in that way is shrinking. Second, industrial development has also shrunk the natural habitats which were existing or available for the wild to survive. Human activities, constructions, development, and reclamation of the land by humans for its own purposes have actually claimed so-called wastelands, which were the houses for the wild species to survive. So if humans claim those lands, naturally their habitats are lost. They move away from that, and if they move away from that, they occupy some other space, which might be the space of living for some other species. So there is aggression happening across the habitats of one species over the other, and that creates the conflict between the wild species also. Let aside the human wildlife conflict, there is conflict among the wild species also for survival, and that is also sometimes one of the reasons why the wild animals or wild species are in danger of uh, uh, being extincted. Over the centuries, we have seen that several species have been removed from the surface of the globe and even under the seas. So this is because of many reasons that I was narrating, and we generally have a feeling that any species that is in danger must be conserved. Now this is a very big issue. Can we conserve each and every species on the earth and we make available all the resources for all the wild animals and wild species on earth? Is it possible? It is a surmountable task. It is unsurmountable task. We cannot do conservation and captive breeding of each and every species. You can never think of allocating the resources to all the wild species that is existing in the globe. So we have to set the priorities, which to conserve and which not to conserve. Taking the thread from what Ms. Karkaria was telling, there are certain species which have innate number of population, number of individuals in a population, which may not be requiring conservation, because for survival of any species, innate abilities of the species to withstand the stress of the environment is important. How best it can cope with the threats naturally, that is one thing. How best it can combat with the diseases which it encounters, either from other wild species crossing the species barriers or coming from the domestic population in the peripheral area and creating a threat in the population. So how it can protect itself from the threats of the diseases, how it can naturally reproduce. What is the generation interval, the generation gap between the generations in a species, how fecund the population is, what is the rate of reproduction of a particular species, what is the number of say offspring a particular mother produces in a given breeding season, and what is the rate of survival of the infants in the natural environmental conditions. This all decides which number is sufficient to feel that now it is a safe species. So IUCN, declares the populations of wildlife and what is the number, and declares the status, and there is a color code for declaring a particular status in certain zones. It says that certain animals, say certain wildlife is in danger, endangered, or it is at risk, or it is less at risk, or it is free from risk. So these numbers also vary from species to species, and there is not a fixed number that if any species is having, say, 500 numbers, it is out of danger. Some species 500 number is out of danger, some other species 500 number is very much a risk. It depends upon the basic physiology of that particular species, basic reproduction parameters of that particular species, and the sustainable environment under which the wildlife is being reared or it is living. So there are several factors which decide whether to conserve or not. On the larger scale, every living species on earth need to be conserved, need to be protected, but we cannot spend money on each and every species. That's why the governments across the globe have their own agenda of choosing the species which requires support of captive breeding or conservation efforts. Now coming to the captive breeding, not all species which requires to be conserved require captive breeding. It is not necessary that any species which is in danger is put under captive breeding program. There are certain requirements for captive breeding. Say we need to imitate the climate or environment under which a species survives in nature. So we need to replicate that kind of environmental conditions in captivity. So this is the most important, the biggest challenge for the people who are involved with conservation biology. It is very difficult to imitate what is exactly available to the animals in the wild. Even then, maximum efforts are made to imitate the nature and recreate the nature in captivity. With limited resources, we make the efforts to copy the natural environment under the captivity. We have to provide right climate, right feeding or food material, and protection that is necessary for survival of a species in captivity. So captive animal management is very important aspect. I will not deal with the captive animal management aspect because that is what has been discussed, and some other people who are to talk on the issue will talk about the management of the captive animals, and they will also talk about the management of the or control of the diseases under the captive population. What I am interested is to talk about the captive breeding and then reintroduction of the species. For each species, there is a species survival plan, SSP, devised under the guidance of the national authorities dealing with the wildlife and the international organizations like IUCN who are involved in the conservation efforts of wildlife. So coming to captive breeding, in conservation, there are 2 or 3 ways in which we make the conservation. In situ conservation is one way where we protect the natural habitat in such a way that there is minimal damage done to the natural habitat of a given species. We closely monitor the aggressions or human activity occurring surrounding that habitat. We minimize the human intervention into the habitat, and see that all the natural resources which are necessary for survival of a particular species are maintained as such without much effect. The only effort that is made is the effects which happen because of the major changes in the infrastructure surrounding the natural habitat, which are inevitable because of the human population. So any ill effect, suppose the building of a particular dam in a given region will affect the wildlife which survives in the downstream of the river where the dam is built. So wherever such kind of developmental activities are inevitable, then relocation or making maximum effort that the species which survived on the banks of such kind of downstream of the dams, they are protected as much as possible, and also trying to make up with whatever changes that happen because of the climate change. We cannot do much as regards to climate change. Only we can make the effort that the minimal damage of the climate change happened in the natural habitat. Floods and famine, these are the two biggest dangers to the habitat of wildlife, whether it is Asiatic lion or rhino or elephants or tigers or primates and amphibia or the birds. Everywhere floods and famine are, these are the two biggest threats to the conservation efforts. So in situ conservation is the best approach. Make best efforts to see that a wild species survive in their natural habitat itself. If we fail to protect the natural habitat, then only the question of ex situ conservation comes into action. Now, there are two ways of doing ex situ conservation. Either you relocate this species in a similar environment outside the natural habitat, which may be similar to the natural habitat, and under the relocated environment or region, we provide all the necessary uh, the inputs which are, we ensure, rather not provide, but we ensure that all the necessary inputs which are required for survival of a species are available there, and additional protective measures which, if required, may be provided. This is the case where the people are talking about translocating additional lions to the regions such as central Gujarat, central India, that is Madhya Pradesh, and other regions. So relocation is one way of doing the conservation in the natural environment, that is ex situ conservation. Ex situ, it is called ex situ because it is not in the original habitat of a particular species. So it is relocation. So in that way, it is ex situ conservation. The second ex situ is keeping the wild animals in captivity in the zoos. So this is also an ex situ conservation because we remove the animals from their natural habitat. We rear them in the cages or in the uh, or the large open cages, and then provide the food and protection from diseases in the zoo conditions, keeping in mind the natural requirement of a species, and we provide all the necessary inputs and protection for that particular species. So this is another ex situ conservation. And the last ex situ conservation is about the conservation of the biological material of the species concerned. Biological material may be the cells of the body, maybe eggs, maybe spermatozoa, maybe DNA material, maybe tissues of a particular species. So these are different ways in which we may make the conservation. The captive breeding is the aspect that is part of the ex situ conservation in the zoos, where we provide necessary support for survival of a species under captivity. When it is captive environment, we must ensure that what is naturally required for a species in the wild is provided in the captivity. When we do this, there are certain dangers or certain limitations. One of the important question that comes is how much to rear in the captivity, how many animals required to be managed under captivity. We cannot keep every animal under captivity and breed them and then again release them. So that is involving cost and managemental expenditure as well as manpower. It is not possible. Limiting the limited resources are available with the wealthiest of the country also. So that is a big issue. So availability of resources and money for conservation decides the captive breeding plan. How many to kept under the captive breeding program. Second is, what is the disease threat under captivity? The animals which are under the natural environmental conditions may not be likely to contract diseases from domestic animals or other wild animals when they are moving around in the wild in their natural way. Once they are put into captivity, they are under the risk of contracting the diseases which are prevalent in domestic animals. If they are zoonotic, they are likely to enter into the wild population. Secondly, they are also likely to gift some of their diseases to other animals and even human beings. So this is second risk. Another issue is providing the administrative continuity in the setup for conservation of the animals in captivity and running the captive breeding program. As you know, there is always the paucity or limited number of experts available in breeding, and therefore limited number of people available for captive breeding program. Conservation of biologists are in limited number, resources are in limited number, and therefore we cannot have very large human resource devoted to captive breeding. We do not have enough number of experts also to perform the activities of captive breeding. We do not have technical support mechanism to train the conservational biologist in advanced technologies such as conservation genetics and genomics and DNA technologies related to the assessment of the diversity of the captive population and the wild population at large. So there is limitation of the technical manpower also to do this. So keeping everything in mind, we need to decide what should be the number that can be sustained under the zoo conditions. Second risk is the quality of the feeds which are provided in the zoos. One important change that is likely to happen under the captive breeding is that knowingly or unknowingly, the human interaction and human intervention happens in the breeding program. Ideally, there should not be any selection happening in the wild, and natural selection only should operate. No artificial selection should happen. No personal choices of the zookeeper or the conservational biologist should enter into the process of choosing the mates to produce the next generation. Choosing the mates for propagation of the next generation is called the process of selection. We as the animal breeders are fond of doing this thing in domestic animals. I, as an animal breeder, breeding the domestic animals, is well trained to choose the animals, selection of animals, and allowing a certain part of the population to propagate in large number is compared to the other part of the population. In a way, I would like, as a breeder, any breeder would like to see that larger and larger proportion of the next generation exhibit or have certain desired traits or characteristics expressed in the next generation. And that way, the breeder wants the population to change over generations in desired direction. And the breeders are trained to do the process of selection precisely and accurately, choosing the traits, identifying the preferred traits, identifying the traits which are undesired or unexpected or unwanted traits, and choosing against them, and then propagating only the selected population, allowing them to breed and then to produce or contribute to next generation. So selection is the process that is being dealt with by the breeders in this manner. He exerts selection pressure, limiting the number of mates, limiting the number of males, limiting the number of females, and seeing that the desired character is passed on to next generation. So this is called artificial selection. This is well documented and well designed experimental or say breeding programs or plans are there, and accordingly we have moved our domestic populations in desired manner. This is the factor that is to be avoided in captive breeding program. So your entire knowledge of animal breeding as regards to selection, as regards to intensity of selection, as regards to selection pressure, and the proportion of the mates, that goes into, I will say, I should not use that word, but into a corner or a dustbin. That means a captive breeder has to think of not doing any kind of artificial selection. We generally, as the human, have the tendency to choose, even in the wild animal, if a certain type of mane or the color or the body shape is there, then the breeder would want that the next generation should be similar to the father or similar to the mother. And we praise if the breeder has ensured that the progeny is also similar to the father as far as possible. So these fanciful ideas or methods which are naturally happening into the minds of the captive breeders has to be controlled. That means there should not be any kind of artificial selection happening in the captive population. Now the question comes, there are certain detrimental genes which are likely to be expressed in the captive brood populations because of the inbreeding that happens under captivity, and deleterious genes are likely to be expressed. Should we eliminate them or not? Ideally, we should not be doing any kind of selection, but for the survival of the species and to see that the species sustains itself over the centuries to come, we need to contain the deleterious genes which are expressed at any course of time during the captive breeding program. They need to be removed. So that much part of artificial selection is likely to remain. That will always be there, and we need to eliminate deleterious genes, or what is generally in the common terms called as the outcast of the population. they need not be conserved, they may be eliminated. Certain abnormalities may be there, certain deformities may be there, certain deleterious expressions of the traits, certain inherited disease predispositions which may be there, they are otherwise also naturally going out of the population. So removal of such kind of animals from the population is either way, it is always going to happen. If the species cannot withstand that deleterious gene in the population, it is naturally going to go out, and it will go out fast when the population is released in the wild, because its survival will depend upon how strong the next generation is. So most important point here in captive breeding is determining what is the effective population size to avoid inbreeding. I will not go into the theory behind how to calculate effective population size and what is the sufficient number for a given species. It changes from circumstances to circumstances, population to population, species to species. But to tell a simple thing is that effective population size is very much governed by the number of males and females which are breeding. If you just remember the simple clue, if there is one male mated to 100, and if it is 50 males mated to 50 females, the effective population size in the latter case is very, very high. 50 males mating to 50 females, that is 1:1 sex ratio in a population is higher effective population size as compared to 1 male and 100 females, or 5 males and 100 females, or 10 males and 100 females. So larger the proportion, in a way, more balanced is the gender in mating population, more will be the effective population size. So keeping aside the formula, if you simply remember that we maintain the sex ratio as close to 1 as possible, is ensuring that we have sufficiently large effective population size. Why effective population size is important? Because males and female contribute equally to next generation. So if one of the two sexes is limited in number, it will expand in a greater way as compared to the sex which is in larger number. Say one bull and 99 cows, one lion and 10 lionesses is a disparity as regards to sex ratio, 1:10 or 1:100. But if it is one lion and one lioness, one bull and one cow, it is equal. So they contribute equally to next generation. So anything that is harmful in a particular male is likely to propagate faster if the sex ratio is deviated towards the male side. That is why, say for example, in lion, you may be finding now in newspapers and several articles also that a very large pride of the lion has been observed in a single location. This is not a happy sign. Why? I will explain. When the pride size increases, it indicates that if it is in a single pride, there is one lion, two lioness and certain cubs, its sex ratio is little in balance as compared to a pride of 25-30 animals where one or two males, one or two young males growing, one or two adult males, and certain number of females and cubs. So larger pride means sex ratio is more deviated towards the negative side. Effective population size is likely to be lesser because lion males do not want other lions to remain together in the pride. The moment the male gets adult, either he is killed or thrown out of the pride. So males dominate in the pride and the number of females are there in larger number as compared to the male. So sex ratio is likely to be deviated in the negative manner. So smaller pride size is in favor of higher effective population size. This also is to be ensured in captivity, because lesser the effective population size, greater is the risk of inbreeding. And when inbreeding happens, the deleterious or negative effect of inbreeding will come into play, and inbreeding depression will happen. The vitality, the vigor, the thriftiness, and the ability to withstand the diseases all will go down in the inbred population. So this is the real risk to captive breeding program. We must ensure that the effective population size under captivity are maintained. Mating between the unknown individuals, unrelated individuals, and exchange of the captive breeding population across the captive breeding programs, across the zoos is very important, exchange of the males, and seeing that there is no like to like mating happening as the breeder or the zookeeper wants. Zookeeper always wants a certain characteristic to remain exhibited in a particular species. But theory says that we must breed rather than like to like, we must breed for unlike to unlike. And we must translocate the breeding animals from one place to another across the zoos where the captive breeding program is there, to ensure that the inbreeding depression do not happen, inbreeding is contained, and effective population size is maintained in a large number. And because if we do not ensure this, the population which is under captive breeding is likely to suffer from diseases, might have lower ability to withstand the diseases, lower immunity to diseases, may contract new diseases which otherwise were not occurring in a particular species, and the population is sometimes in danger of uh, reducing the population because of the disease threats. Sometimes the entire population may be wiped off in this entire zoo. So this should not happen. Regarding reintroduction of this captive bred population, I will not go into the details of measuring the diversity of the captive bred population through the use of modern genetics technologies like microsatellite markers, taking the samples, assessing the microsatellite diversity, and assessing the genetic relationship or kinship among the captive bred population, and then going ahead or using the genomics method for assessing the S&P variation at large number of loci, and then going ahead. So I am not dwelling into this highly technical issue, and I will limit my talk. I will limit my talk only now to cover the part of reintroduction.
Sir, 10 minutes are remaining, sir.
Yes, yes. I will finish within 10 minutes because I also have another assignment. Okay. Yes. So now coming to the reintroduction. Now, as you know, under the captive breeding plan, the population is not being reared under the natural environmental conditions. So when the population is not reared under the natural environmental conditions, say for example, lion cubs born in Sakarbagh Zoo, they have never seen the natural Gir sanctuary conditions. What they have imitated is just in the zoos. So certain natural training that happens to the cubs in the natural habitat never happens to them. So certain things they never learn. They never learn how to scavenge for food, how to reach for the waters, how to protect itself from the naturally occurring other wild species, and how to they survive under the sun. How to protect itself. So they do not learn. So how to make them learn is the biggest challenge. If they do not learn, and if they are released in the wild, they are domesticated wild animals. So natural wild animals and domesticated wild animals do not behave the same. So if we are planning to reintroduce the captive bred population into the natural habitat, we must train them to imbibe into qualities which are required in the natural conditions. So there are two types of releases. One is called as hard release, another is called as soft release. Only the processes are different. You will find the process of soft release and process of hard release in literature. I will not go into the details. So release plan has to be decided. Which release plan is important for a given species? Before release, what kind of training is required, we try to give. But one thing is certain, captive animals are getting their food coming from the slaughterhouses. They don't have the training for hunting. Even if artificial hunting facilities are provided in captivity, the prey is not free to protect itself. So it's an easy game for the wild animal to prey on the herbivore on which it is hunting, and it is just a very safe game for the wild animal to hunt on the animal. So this is not giving them the real training that is required in the wild. Natural instinct of attacking. One observation that was observed in one of the wild species was that when the captive bred animal was released to prey on a particular prey animal, rather than approaching the neck region to press the jugular vein, the attack was made on the back of the neck by the captive bred wild animal. This is not going to kill the prey. If it is not going to kill the prey is like to escape. So the skills which are naturally required, for hunting, if they are not learned by them, they will not be able to survive, because they will not be in position to feed on the prey, and they will fail. Similarly, certain traits of racing and then capturing the hunt, capturing the prey, and then making that fall, and then catching the neck. All these process are the natural skills that the animal learns in the natural wild condition, which is not possible in the captive breeding plan, captive bred population. So some kind of training is required, otherwise the animals will be shy. Rather than wild, they are domesticated wild animals, and they will survive less. So this is one important component in reintroduction. Second important component is, wherever the reintroduction is happening, natural population is always supposed to be there. So there is a possibility of conflict between the reintroduced animals with the naturally living wild animal in that particular habitat. For them, it will be strangers. What we see in the street dogs, if the street dog in my area moves to another street, then the local dogs of that particular street will be attacking on the dog of another street, and it has to run away. It may be killed or it may be injured, and it has to run away. Similar kind of conflicts happen between the reintroduced species with the natural species that is existing in the wild. Especially in lions, this is likely to happen because there is a strong priding behavior that is seen in the lions. That is naturally in most of the felids, they attack each other, and they do not allow the other wild animal to enter into what is called as the zone that the animal decides that this is my zone, this is my area, this is my habitat. So they demarcate the territory, and nobody is allowed to enter into the territory. So how to make the reintroduced population to enter into the territorial boundaries of the natural wilds, that is also an issue. That's why soft release is necessary. How they become familiar, they become part of the pride of another wild animal. This is very important. They should also be able to form their own pride and sustain in a new territory that they capture, and then protect the territory, because even in the natural wild population, the young adults and the old adults, they fight with each other, and stronger will survive, and survival of the fittest plays a great role in the natural conditions also. Here the captive bred animals will be at the receiving end, and they are likely to be killed, because the another natural bred animal will be having stronger traits, stronger abilities, stronger skill sets with it to fight with the captive bred animal, and the danger is always there. So there are several factors which are important in reintroduction of the species. There is cost of reintroduction also, because we need to give extra protection to the reintroduced population for some period of time to see that such kind of conflicts do not happen. Another important point that should be brought home in the mind is that so-called domestically bred, that is captive bred wild animals, have seen lot of human interaction in their lifespan before they are reintroduced. So they are more inclines to be with the human beings rather than other wild animals. So there is a possibility that they will rush towards the human habitats more faster or more quickly, more easily than the naturally wild living animals. They will remain in their area only. These animals have the tendency to move towards the human population. So this again creates the threat in the public, that the certain zoo authorities have released the lion in a particular area, and that has entered the village. Certain leopards are released in the wild, and it has now started attacking the human population. So this is a real threat. So there is a need to make public aware in the about the process of reintroduction happening in a particular area. Public education as regards to the conservation efforts made, to captive breed the animal, and efforts made to reintroduce the species into a particular area, so that people don't go against the program. So population conservation is one aspect, and the society education is another aspect that is also important in captive breeding program. Captive breeding program and reintroduction program as such do not demand the social work, but for sustenance of the program we need to do the social work and public education as regards to release, and then ensure that people also favor this kind of effort. I think I have dealt with almost all the aspects of captive breeding and reintroduction. I have kept deliberately the technical discussions and details of captive breeding and reintroduction process out of my speech, because that is tremendous literature available on these aspects, and I wanted not to repeat that. I wanted not to again take a class and teach that, but I wanted to interact with you on some important salient features of the captive breeding and reintroduction, so that we ignite the young minds and they are prepared well to think about the need of captive breeding, especially for the endangered wildlife. There is a great career that is ahead for an interested veterinarian who is interested to devote his life to captive breeding of wild animals. And as you will see in the entire 5 days training program, there are several areas where the veterinarians are required, but the conservational biologists who are working with the zoos have not realized the importance of veterinarians, except in treatment of the animals. There is a greater role of veterinarians other than only treatment of the animals in various aspects of zookeeping and captive breeding, reintroduction, and relocation or translocation or in situ conservation as well as ex situ conservation. Especially when the population is danger, the veterinarian has a greater role to play than the people who are working with the zoos in normal conditions. In normal conditions, it is very easy to maintain, but when there is a threat, veterinarian is always the first man who can support a species and support the mechanism for conservation of a wild species. So with these words, I thank once again the organizers, Dr. Anshu Helawat, the entire team of organizing team, and the patron, Vice Chancellor, the Director of Research, the Dean of the faculty, and all those who have participated and listened carefully to what I have spoken at length. So I have taken more than the time that was expected. Thank you very much.
Thank you very much, Dr. Vatalia, sir. You have nicely presented regarding this captive breeding. Dear students and friends, is there any question to our respected speaker? So I think that there is no question, Vatalia, sir.
I have deliberately I have avoided the PowerPoint presentation and going into the formulas and methods and all these things, just to keep it a formal talk so that you people can ask the questions. But anyway,
you have presented in traditional manner, sir. You have presented in traditional manner.
that was my objective.
Sir.
That was my objective, not to teach, but to talk.
Yes. Sir, what about your planning regarding this opening of any wildlife research center in Kamden University? After integration of colleges or
day before yesterday? Yes, you are making the right point. Day before yesterday, we had the research council meeting on 15th, and at that in that particular meeting also, I told that Kamden University is interested in starting a wildlife research station under Kamden University. Under Kamden University doesn't mean at Gandhinagar, doesn't mean at the present locations under control of Kamden University. So Kamden University has open mind wherever wildlife is there, wherever the most feasible location is possible. Kamden University in future is interested in having a very good wildlife research center entirely devoted to wildlife health, wildlife conservation, and providing all similar services which are required. Remember, wildlife is not only lions, not only tigers, it is not only alligators and gadial, it is n number of bird species, n number of aquatic species, and the amphibians. So where to concentrate is the choice of the people who will man such kind of wildlife research station. So enormous possibilities are there, and especially in wildlife medicine, veterinary colleges can contribute a lot. We have seen that in the past also, and we'll see that in the future also. Thank you.
Definitely, sir. After integration of colleges, you are having a very dynamic leadership, and I think that there will be really development if any center is open in Kamden University. So, dear friends, we had a very respected speaker and eminent speaker, Dr. Vatalia sir. He had narrated each and every point regarding this captive breeding, translocation, and what about the soft and hard release in the reintroduction, and nicely presented in a very simple way. So I again thankful to you, sir, on behalf of your close friend, Dr. P.H. Vatalia, sir, and all organizing team, sir. Thank you very much, sir. Thank you very much for joining.
Your screen is so small, I don't recognize you, mister.
I'm Dr. Savarkar and Dr. Flipper.
Oh,
sir.
Okay, Dr. Savarkar. I just from the your accent, I could make. I was just doubting. That's why. Dr. Ansu Mahalabat, I can see.
Yeah.
See, wherever you are sitting, there is no enough light on you. So make some arrangements. So whatever limitations are there, but I thank you very much for giving me this opportunity. I will sign off. Thank you.
Okay. Okay, sir. Thank you very much, sir. Namaskar, sir.
Thank you. Thank you very much, sir. Dear student, we have another very good speaker, Mr. Yagnesh Bhatt. First of all, I brief give the introduction about the Yagnesh Bhatt. Before that, first of all, I welcome Mr. Yagnesh Bhatt, sir, on behalf of the our team and the colleges. Welcome, sir. Myself, Dr. D.T. Pepper, and
thank you.
He is Dr. Savarkar, sir. Okay, he is the chairman of this session. My voice is audible to you, sir?
Yes, yes. I am audible
okay,
to you?
Yeah, yes, sir. Yes, sir. Yes, sir.
Fine.
So, with brief introduction, Mr. Yagnesh Bhatt, sir, did his graduation in the chemistry, and after that, he has also completed his master degree in the philosophy. But in spite of his interested in philosophy, but he is very interested in the philosophy of the wildlife. Okay, he has spun in the several years in the pharmaceutical company, but apart from them, he is very good, very much a lover of the wildlife, and he has done very good job for this wildlife conservation. And he has also published the number of his papers, especially in the BBS wildlife calendar as well as the magazine. Okay, and he is the permanent member of the Mumbai Natural History Society, as well as he is also a permanent member of the Conservation Society of the Gujarat, Bird Conservation Society of Gujarat, that is a BCSG. So with this brief introduction, now I request Pankaj sir to give some technical, or we can say give some throw lights on the bird rescue and rehabilitation. Please, sir.
Okay. Please allow me to share my screen.
Yeah. Sir, now you can share your screen, sir.
Yeah. Okay. I am very thankful to you all, Department of the Animal Genetics and Breeding, Veterinary Science College of Junagadh, NAHEP and IDP, Dr. Tak Saib, Dr. Rak Saib, Dr. Allahabad Saib, थोड़ा मैं हिंदी गुजराती इंग्लिश का यूज करूंगा, तो मुझे टॉलरेट कर लेना। एक्चुअली हमारे जो विद्यार्थी है और जो स्टूडेंट्स को जो मैं एड्रेस करना चाहता हूं, तो थोड़ा बहुत मैं जो यहाँ की हमारी पद्धति है, आपने अभी तक मैंने देखा कि अभी तक आप जो इंफॉर्मेशन, टेक्नोलॉजी, टेक्निक्स, स्टैटिस्टिक्स सबके अंदर जो आप उनके अंदर का जो इतना इंफॉर्मेशन, इतना एजुकेशन के साथ आप चल रहे हैं, मगर मैं थोड़ा आपको डिफरेंट एक ट्रेल पर ले जाना चाहता हूं, कि जो ट्रेल पर मैंने बचपन से आज तक सफर किया है। एक्चुअली मेरा जो बचपन है उसमें से तब से मुझको अटैचमेंट था नेचर, बर्ड्स, एनिमल्स और सब आसपास का जो नेचर था उससे मुझे बहुत लगाव था। और छोटे बचपन की जो आदतें होती है, जैसे स्कूल में जाते हैं तब मुझे अभी याद है कि जब स्कूल में जाते थे तब छोटे छोटे जो क्रीचर्स नेचर के हैं वो हमको मिल जाते थे मैदान में, हमारे खेलने के मैदान में, तो उससे मैं बहुत अट्रैक्ट होता था। और बाद में उनमें जो मेरा रुचि बढ़ने लगा, तब कई बार जो नेस्ट से ड्रॉप आउट हुए बच्चे हैं, स्पैरो के या फिर मैमल्स में गिलहरी के, बर्ड्स में पैराकिट के, तो तब मुझे ये रुचि रहती थी कि उसको मैं रियर करूं। मेनली जो मैंने अभी तक का जो पाया है ना, उसमें मैं ये कह सकता हूं कि जो इंट्रोडक्शन होता है ना, जब किसी से भी, विदाउट इंट्रोडक्शन उसके प्रति प्यार नहीं होता है। तो मैंने वही अपने जहन में भरकर रखा है कि जैसे मैं जितना जितना मैंने जो किया है वो मैं सबके साथ शेयर करता हूं, और जो मैंने पाया है उनसे वो शेयर करता हूं, जिससे इंट्रोडक्शन हो जाए। और आज जो स्टूडेंट्स है वो बिलीव करेंगे ही कि प्यार हो जाए बाद में क्या होता है सबको मालूम है। उसके लिए क्या करना, क्या नहीं करना, क्या कर सकते हैं, कितना डेडिकेशन से कर सकते हैं, वो हमको मालूम है। तो मैं इसलिए ही यही एक फ्लो में जा रहा हूं कि ये जो बर्ड रेस्क्यू एंड रिहेबिलिटेशन, एक्चुअली आज की जो हमारी लाइफ है, और आप जैसे वेटरनरी स्टूडेंट्स हैं, उनकी लाइफ में तो भर भर के बर्ड्स एनिमल्स आने वाले हैं। तो उनके साथ का जो आपका रिलेशन, उसको समझना, उसका इंट्रोडक्शन, तभी जाके उससे प्यार होगा। और मैं आपको मेरी लाइफ से एक बात बताता हूं कि when if you are fall in love with somebody, तो जो उसका हमारा तरीका आता है उसके साथ डील करने का, वो कई अलग तरीके से आता है। क्योंकि एक्चुअली जब हम वो क्रीचर की महत्वता समझते हैं, अभी तक सब लोगों ने, सब प्रोफेसर्स, ये वाटलिया सर ने भी इतना बढ़िया सा समझाया सब कुछ, कि जो टेक्निकलिटी है, टेक्निक्स है, और जो साइंस है, तो वो बहुत सारा आपको मिलने वाला है। आप जब 4 साल का या 5 साल का आपका जो स्टडी है, उसमें ये सब बहुत मिलने वाला है। मगर वो जो मिलेगा उसको यूज क्या करेंगे, उसको कैसे हम हमारी प्रैक्टिस में लाएंगे, तो उससे आपको बहुत फर्क पड़ने वाला है। जो मैं ऐसा समझ रहा हूं, जो मैंने समझा है, एक्चुअली मैंने जब स्टडी किया, मैं बचपन से ये आदत मेरी थी, मेरे फादर डॉक्टर है, तो मेडिकल का थोड़ा नॉलेज था। मैं फार्मा इंडस्ट्री में रहा, तो फार्मा का मुझे बहुत नॉलेज मिला। तो जो ह्यूमंस का जो नॉलेज था, उसको ही मैंने ज्यादातर अप्लाई किया, क्योंकि तब मेरे पास इतना स्टडी करने का ऑप्शंस नहीं था। आज तो इतना स्टडी का ऑप्शंस आ गया है, यूट्यूब है, गूगल है, एवरीथिंग। तब मेरे पास ये सब नहीं था। तो मैंने धीरे 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 करके ये सब टेक्निक्स को डेवलप करने का प्रयत्न किया है। तो मैं आपको आज वही टेक्निक्स की, जो मैंने बर्ड रेस्क्यू एंड रिहेबिलिटेशन में क्या क्या प्रयोग किए हैं, उसमें से क्या मैंने पाया, क्या किया, तो वही जर्नी पर ले जाना चाहता हूं। एक्चुअली आज हम जो बात करेंगे उसमें जो कई अलग अलग सब्जेक्ट पर बात करेंगे। तो इंट्रोडक्शन करेंगे, रेस्क्यू करना कैसे करेंगे बर्ड को, उसको वाइल्ड में कैसे ले जाएंगे। अभी वाटलिया साहब ने बहुत बढ़िया बताया कि जब वो कैप्टिव ब्रीडिंग हो जाता है या फिर ह्यूमन टच आ जाता है, बाद में बहुत प्रॉब्लम आता है वो बर्ड या एनिमल्स या क्रीचर को। तो वो हम कैसे कर सकते हैं, जो मैंने थोड़ी बहुत टेक्निक डेवलप की है। हमारे जो एफर्ट्स है, आप तो वेटरनरी डॉक्टर होने वाले हैं, तो आपके पास इतना सब कुछ नॉलेज आएगा। तो वो नॉलेज आज की तारीख में टेक्नोलॉजी इतनी बढ़ गई है कि कितना भी प्रॉब्लम, इलनेस आ जाए, इंजरी आ जाए, उसको ठीक करने के लिए प्रोस्थेसिस वगैरह सब कुछ डेवलप किए गए हैं। तो उससे हम कैसे उसको अपने जो नेचुरल एनवायरनमेंट, उसका जो नेचुरल पॉपुलेशन है, उसके साथ हम उसको रीएस्टेब्लिश करें, उसको हम रिहेबिलिटेट करें। तो ये सब बात हम देके उसको हम एक सेकंड चांस दे सकते हैं बर्ड को। आज हम देख रहे हैं कि बर्ड इज अ वेरी डेलिकेट क्रीचर, तो उसको हम कैसे ट्रीट करें। पहले तो जब भी कोई बर्ड आता है हमारे पास, या फिर कोई ईल बर्ड आता है, या हमको कहीं कहीं से भी इंजर्ड बर्ड मिल जाता है, आपको मैं बताऊं कि कुदरत ने ह्यूमन को बनाया ही इसलिए है। आप अगर देखेंगे ना, तो कितने बर्ड्स है कि वो ह्यूमन के पास आके ब्रीडिंग करते हैं। कितने बर्ड्स है, स्पैरो, सनबर्ड्स, वो हमारे नजदीक के एनवायरनमेंट में आके ब्रीड करते हैं, क्योंकि वो सेफ्टी फील करते हैं। वो हम जो समझ जाएंगे ना, तो बहुत उसमें से मिल जाएगा हमको मटेरियल। तो ये जो बर्ड है, तो बर्ड जब भी इल होता है या फिर सिक हो जाता है, तो या तो एक कॉर्नर या फिर आइसोलेशन करने का सोचता है, या फिर जो ह्यूमन पॉपुलेशन है उसके नजदीक में आ जाता है। उसको थ्रेट जो अदर क्रीचर से होती है, उससे बचने के लिए हो, या फिर हमसे कोई हेल्प मिल जाए, वो भी हो सकता है। मगर एक जो बात है, वो एक बर्ड है, वो बर्ड को हमारा मेन कंसर्न ये होता है कि हम अनहिल, इंजर्ड, इल्ड और ऑर्फन, ऐसे जो बर्ड्स है, उसको हम कैसे ट्रीटमेंट दे, उसको कैसे हम उसका रेस्क्यू करें। तो वो रेस्क्यू महत्व इसलिए हो जाता है कि जब भी उस रेस्क्यू करते हैं, तो वो केज में ना चले जाए, वो अपनी लाइफ जो है वो नेचुरली बिताए, ऐसा उसके पास ऑप्शन रह जाए। दूसरा एक रीजन है, तो हम उसको, बर्ड को जो भी हम रेस्क्यू करने के समय तकेदारी रखते हैं कि जिससे उसको हर्ट ना हो जाए, उसका क्योंकि वो बहुत सेंसिटिव होते हैं, उसका कुछ भी ट्रीटमेंट करते समय उसका या तो डेथ ना हो जाए या इंजरी ना हो जाए। कई बार मैंने ऐसा पाया है, क्योंकि मैंने जितना जितना काम करता गया तो कई अनुभव होने लगे। तो उसमें से मैंने ये पाया कि उसको सेंसिटिव बहुत होने के बाद उसको हम एक एनवायरनमेंट दे कि जिससे वो अगेन इकोसिस्टम में हम रिहेबिलिटेट कर सकते हैं। सेकंड थिंग, ये बर्ड इतने इंपॉर्टेंट है, जैसे हम लोग सुनते आए हैं फ्रॉम स्पैरो टू वल्चर, इतने इंपॉर्टेंस है उसका कुदरत में रहना। उसका क्लीनिंग, जो क्लीन एनवायरनमेंट रखने का काम है वो तो है ही, मगर हमारे आसपास का जो सराउंड है वो लाइवली बनाने का भी काम कौन करता है? वो बर्ड्स ही ज्यादा है। आप देखेंगे कि विदाउट एनी बर्ड, विदाउट एनी साउंड ऑफ बर्ड, हमको कैसा लगेगा? तो ये बर्ड का इतना महत्व है, तो हम इतना उसका, जब उपयुक्ततावादी समाज हो गया है, तब हम इतना तो समझा सकते हैं और समझ भी सकते हैं कि इसका इंपॉर्टेंस बहुत बड़ा है रखा है। तो उसको इसलिए भी हमें रेस्क्यू करना है, रिहेबिलिट करना है। और रेस्क्यू एंड रिहेबिलिटेशन ये पार्ट है कंजर्वेशन का। अगर वो कंजर्वेशन हम करना है, तो ये पार्ट हमारा मजबूत होना चाहिए। और दूसरा, ये जो बर्ड्स है, ये बर्ड्स में जो एंडेंजर स्पीसीज है, वो एंडेंजर स्पीसीज के लिए भी इतना ही बहुत महत्वपूर्ण हो जाता है कि उसको हम रेस्क्यू करें और रिहेबिलिटेट करें। ये अभी हमारे पास कोई भी एक आध बर्ड आ गया, जैसे ये आप पिक्चर में देख सकते हैं, इट इज अ ब्लू टेल बी ईटर्स चिक. एक्चुअली इट इज ड्रॉप फ्रॉम इट्स नेस्ट. उसका जो नेस्ट होता है वो होल होता है, जो मिट्टी की दीवार होती है जहां से ट्रैकिंग होता है मिट्टी का, वहां वो नेस्ट करता है. तो कई बार फ्रॉम टेकिंग फूड फ्रॉम पेरेंट्स वो कई बार नीचे आ जाता है खिसकते खिसकते. तो सिर्फ इतना ही है कि हम एक छोटा सा हेल्प करें उसको अपने नेस्ट में उसको अगेन प्लेस कर दे, तो हमारा काम बहुत कम इजी हो जाता है. मैंने आज तक जो टेक्निक्स डेवलप की है उसमें मैंने मेनली ये किया है कि मैंने जो समझता गया, बर्ड्स को मेनली चिक जो ऑर्फन मिल जाते हैं या फिर कोई नेस्ट से नेस्टलिंग होता मिल जाता है कि नेस्ट से निकल गया है या फिर नीचे आ गया है, गिर गया है, तो उसको मैं उसके नेस्ट को ढूंढ के फिर उसको प्लेस करना, वही मेन हमारी टेक्निक रहेगी. मेन जब भी कोई भी बर्ड मिला, अगर उसका कोई नजदीक का हमें टेक्निक नहीं मिला कि उसको कैसे हम प्लेस कर सकते हैं उसके नेस्ट में या फिर कहाँ का है कैसा है, तो हमारे पास एक ही काम है कि उसको एरी बॉक्स में प्लेस करके उसको थोड़ा स्पेस दे, उसको टाइम दे. क्योंकि उससे क्या हो जाता है कि जब भी हम उसको टाइम देते हैं तो उसका जो स्ट्रेस है वो रिलीव हो जाता है और जो अपने आप को इंजर्ड भी नहीं करता है और हमारा काम जो है वो इजी हो जाता है. तो मेनली क्या करेंगे, पहले तो कोई भी बर्ड मिला तो उसको हम एक एरी बॉक्स में प्लेस करके फिर आगे का सोचने का. मेन अप्रोच जब करते हैं बर्ड के पास जाने का, तब ये मेन हमारा गोल रखने का कि वो हमारा प्रेजेंस से उसको थ्रेट नहीं लगता है ना. वी हैव टू टेक केयर फॉर इट. थोड़ा उसका कॉन्फिडेंस डेवलप हो जाए, बाद में उसको कैच करना वो महत्व हो जाता है. क्योंकि ड्यूरिंग कैचिंग द बर्ड हम इंजरी ज्यादा कर देते हैं. तो ये महत्व हो जाता है कि हम उसको कैसे कैच करते हैं और प्लेस करते हैं. मेनली छोटा बर्ड है तो हम क्लोथ से या टॉवेल से कैच कर सकते हैं. बड़ा बर्ड है तो मेन हम उसके आंख पर एक क्लोथ डाल के उसको थोड़ा काम कर देते हैं, थोड़ा उसका स्ट्रेस रिलीव करके फिर उसको प्लेस करते हैं बॉक्स में. और मेनली डार्क जहां पर हो उसको प्लेस करेंगे तो इट इज वेरी इजी फॉर बर्ड टू रिलीव द स्ट्रेस. कई बार ऐसा होता है कि ये रेस्क्यू में कैसे कैसे आते हैं बर्ड, तो ये जो कार या व्हीकल से टक्कर हो जाती है या उससे स्ट्राइक होने के बाद थोड़ा बहुत इंजरी होता है. तो मेनली इफ वी पुट असाइड द रोड और उसको थोड़ा सा एक साइड पर रख देते हैं थोड़ी देर के लिए, तो भी उसका स्ट्रेस रिलीव होके फिर वो अगेन फ्लाई कर देता है अपने नेचुरल एटमॉस्फेयर में. तो थोड़ी ही हेल्प, वेरी लिटिल हेल्प कई बार बहुत काम करती है. तो बर्ड के बारे में मेन ये है कि उसको मेन टेम्परेचर मेंटेन करना, ज्यादा बहुत वार्म या बहुत हार्श सन है वहां पर नहीं रखना, उसको थोड़ा काम मिले ऐसा एटमॉस्फेयर रखना. एंड देन find out some organization which is useful for bird rescue and treat them. अभी वेटरनरियन डॉक्टर्स का बहुत बड़ा रोल हो रहा है और इतना अच्छा काम हो रहा है कि मैं देख रहा हूं कि ये जो वाइल्ड बर्ड्स और एनिमल्स के बारे में उनका नॉलेज भी बहुत अच्छा हो रहा है, तो उससे जो विलेज, सिटी एंड फॉरेस्ट के जो बर्ड्स है उसको सेव करने में बहुत हेल्प मिलती है। मेनली काइट फेस्टिवल्स और ऐसे टाइम में बहुत ही एक्टिव रहता है, मगर जब भी, क्योंकि अभी फैसिलिटीज इतनी हो गई है कि उसके ब्लड एनालिसिस और एक्सरे ये सब से इतना इजी हो जाता है वर्क करना, कि इससे बर्ड रेस्क्यू वो जो टास्क था, पहले मुझे याद है कि जब मैंने बहुत साल पहले एक बार मेरे पास एक सिकरा, उसका विंग का बोन फ्रैक्चर हो गया था, तो मैं उसको लेके आनंद वेटरनरी कॉलेज में गया था। तभी उसको बहुत ट्राई किया था हम डॉक्टर्स ने, मगर तब एक डॉक्टर थे जो इंटर्न थे, और मैंने जब वो मैं हैंडल कर रहा था, तभी डॉक्टर जानी साहब ने कि डॉक्टर साहब ने कोई डॉक्टर थे, उन्होंने बताया कि लर्न द हैंडलिंग ऑफ दिस बर्ड, हाउ ही इज हैंडलिंग। तभी जाके मुझे भी थोड़ा बहुत लगा कि ये टेक्निक्स होती है कि हम हैंडल करें, तो टेक्निक्स बहुत बहुत अच्छा रोल प्ले करता है कि हम कैसे उसको हैंडल करते हैं। तभी बाद में वो डॉक्टर को भी बहुत इंटरेस्ट हुआ बर्ड्स के लर्निंग में, और उन्होंने बहुत बड़ा स्टडी भी किया और अच्छा अभी तो काम कर रहे हैं। तो मेन जब भी आपका जो ये वेटरनरी साइंस की जर्नी है, तो उसमें मेन आप जितना लर्न करेंगे ये सब बात, और अपना दिमाग उसमें डालेंगे, उसके प्रति हमारा जो संवेदना है हमारी वो डालेंगे, तो उसमें बहुत कुछ सीखने को मिलेगा। बर्ड जब भी हम रिहेबिलिट करते हैं, तो मेन वो जो हमें ध्यान रखना चाहिए, वो जो बात है, वो मेनली बात मैं थोड़ा डिस्कस करता हूं कि कोई भी बर्ड हमारे पास आया, लाइक ये पिक्चर में हमारे पास एक येलो फुटेड ग्रीन पिजियन है, विच इज वेरी साई बर्ड। जब भी मैंने ये स्टार्ट किया था, ये बर्ड मेरे पास बहुत सालों पहले से इंजर्ड होके आता रहा है, क्योंकि इट इज ओनली डिपेंड ऑन वाइल्ड बेरीज एंड फ्रूट्स, तो उसको रियर करने का बहुत डिफिकल्ट रहता था, मगर धीरे धीरे उसमें मैंने टेक्निक डेवलप की। मगर ये ऐसे जो ट्रिकी बर्ड होते हैं, तो वो ट्रिकी बर्ड को हम कैसे ट्रीट करें, तो मेन एक बात है कि जब भी हम उसको घर में या फिर हमारे पास जब होता है तब रियर करते हैं, तो उसको जो फूड देते हैं, वो फूड का बहुत मेजर रोल है, क्योंकि हम उसको अगर उसकी जो नेचुरल हैबिट है उससे अलग हैबिट में ले जाएंगे, तो फिर वो वाइल्ड में सरवाइव नहीं करेगा। मैंने जब ये येलो फुटेड ग्रीन पिजियन को रियर किया था, तब मैक्सिमम उसको वाइल्ड बेरीज लाइक पीपल, बनियन ट्री, फिग फ्रूट्स एंड ऑल, इसके ऊपर ही उसको मैंने उसको फूड देने का रखा था। तो उससे क्या हुआ कि जब जाके वो वाइल्ड में अगेन उसको मैंने रिलीज किया, तो वो आराम से सेट हो गया। मैंने 5-6 दिन उसका ऑब्जर्वेशन किया था, मगर वो आराम से एस्टेब्लिश हो गया। तो मेन जो बात है, तो वो मेन है कि हम उसकी एबिलिटी चेक करते हैं कि वो वाइल्ड में सरवाइव होगा कि नहीं होगा। उसके लिए भी मैं एक आध दो जो मेरी इंसिडेंट्स है वो आपके साथ शेयर करूंगा, मगर मेन जो ये ऐसी इंसिडेंट्स है, वो उससे मैं सीखता गया कि कैसे उसको हम रियर कर सकते हैं और वाइल्ड में उसको रीइंट्रोड्यूस कर सकते हैं, उसको वाइल्ड में हम रिलीज कर सकते हैं। कई बार ये अगर नेस्ट बर्ड आ गया, नेस्टलिंग, तो हम उसको रियर करेंगे, मगर फाइनल हमारा जो गोल है उसको रीएस्टैब्लिश करना है वाइल्ड में, तो हम कैसे करेंगे? तो उसको भी यही कि उसका नेचुरल फूड हम देंगे, नेचुरल फूड देंगे, उसकी एक्टिविटीज मेजर करेंगे, क्योंकि उसको विंड फ्लैपिंग करना, उसको उड़ना, ये सब जो हम उसको ट्रेन नहीं करेंगे तो उससे फिर वो वाइल्ड में उसका बचना बहुत मुश्किल हो जाता है। महत्व का दूसरा एक बात ये भी है कि ज्यादा ह्यूमन टच न हो जाए, वो उसका भी ध्यान रखना है, क्योंकि जितना हमारा ह्यूमन टच ज्यादा बढ़ेगा तो उसका नेचुरल इंस्टिंक्ट्स है जो ह्यूमन पॉपुलेशन से दूर रहना या फिर कैच ना हो जाना अगेन, तो वो चला ना जाए। तो इतनी थोड़ी थोड़ी छोटी छोटी बातें जो हम ध्यान रखेंगे तो ये जो बर्ड रेस्क्यू एंड रिहेबिलिटेशन हम बहुत आसानी से कर देंगे। अभी मेनली मैं जो जो मेरे साथ जो इंसिडेंट्स मुझे मिले हैं, मेरी जो जर्नी है, उसमें से आपके साथ थोड़े मेरे इंसिडेंट्स शेयर करता हूं, क्योंकि मैंने एक तो मेरी हिम्मत, क्योंकि मेरा स्टडी नहीं था, मेरा एक प्रकृति के प्रति प्रेम और थोड़ा सा हेल्प, थोड़ा सा मेरा प्रयत्न, उसमें से जो रिजल्ट मिला वो आपके साथ मैं थोड़ा मेरी जर्नी शेयर करता हूं। फर्स्ट जो एक इंसिडेंस लेते हैं वो लेसर विस्लिंग डक एंड डकलिंग्स रेस्क्यू। लेसर विस्लिंग डक वो हमारी नेटिव स्मॉल डक है जो कई बार ह्यूमन पॉपुलेशंस के नजदीक में नेस्ट कर लेती है, क्योंकि अभी उसका जो एनवायरनमेंट है ना, हमने उसके ऊपर जो एंक्रोचमेंट कर लिया है, तो उससे ऐसी सराउंड उसको नहीं मिलता है कि वहां पर वो नेस्ट करे। दूसरी भी एक बात है कि मैं ऐसा कह भी सकता हूं कि उसने ह्यूमन पर थोड़ा विश्वास भी जताया है, उसके बारे में भी बताऊंगा, जो मेरा उसका मैंने जो उसमें से पाया है। तो लेसर विस्लिंग डक वो स्मॉल डक है जो अप्रोक्सीमेट 10 to 12 या 15 एग्स ले करती है। और हमारा जो गार्डन है, आजकल मैंने जो जो देखा है, तो हमारा गार्डन होता है, तो गार्डन के अंदर शुरू के ट्रीज का जो वॉल बनाते हैं, तो उसमें जो कैविटीज बनती है, या फिर जो कोकोनट ट्री की ऊपर जो कैविटीज बनती है दो उसके पत्तों के बीच में, वहां पर वो अपना नेस्ट करती है। लेसर विस्लिंग डक के जो बच्चे हैं अप्रोक्सीमेट अभी तक मैंने 10 to 15 का जो क्लच देखा है, और मेनली मेरे गांव में हर साल 10 to 12 डिफरेंट पैर को गांव के नजदीक में बच्चे रियर करते हुए देखा है। तभी जाके क्या हो जाता है, अभी वो गांव के बीच में उसने नेस्ट कर दिया, गांव के आसपास में जो बंगलो है उसके अंदर नेस्ट कर दिया, तो वो कैसे हम उसको रेस्क्यू कर सकते हैं, क्योंकि ये बर्ड बाय ओनली लैंड ही उस बच्चे को कोई राइस के फील्ड में या फिर फ्लडेड फील्ड में या तो फिर नियर बाय वाटर बॉडी में ले जाएगा। तो हम उसको हेल्प क्या कर सकते हैं? मैंने जो किया है वो आपके साथ शेयर करता हूं। स्टार्टिंग में मैं उसको मेरे जहां पर ये बर्ड के लेसर विस्लिंग डक और डकलिंग्स मुझे मिले थे ऐसे, तो वहां से मैंने क्या किया था, 2 लोग को साथ में लेके उसको गाइड करते करते वाटर बॉडी तक ले गया था। और वाटर बॉडी तक ले जाने के बाद उसका काम इजी हो जाता है, क्योंकि वो वाटर बॉडी के पास आसपास में तभी ये मॉनसून में ही उसका नेस्ट होता है, तो तब ग्रास भी इतना होता है, वो आराम से बच्चे वहां पर हाइड भी कर सकती है और वहां पर रियर भी कर सकती है। कई बार ऐसा होता है कि सिचुएशन ऐसी आ जाती है कि जब भी ये जो बर्ड के डकलिंग्स है, वो डकलिंग्स बिल्कुल बाजार में या फिर बहुत ह्यूमन पॉपुलेशन के बीच में होते हैं, तभी मैंने ये टेक्निक की है कि वो बच्चों को 2-3 बच्चों को मैं पेरेंट के साथ रहने देता हूं और दूसरे बच्चों को मैं बास्केट में ले लेता हूं। बास्केट में लेके आसपास में जहां पर भी मुझे ऐसा कवर्ड एरिया मिले कि जहां पर छोटा सा रूम मिल जाए, या फिर हम यूज भी कर सकते हैं, जैसे अभी मॉस्किटो के लिए हम मच्छर जाली जो रात को सोने समय हमको पॉपअप मिलती है, उसका भी हम यूज कर सकते हैं। उसके अंदर मैं वो बच्चों का बास्केट रख देता हूं और धीरे धीरे पेरेंट और 2-3 बच्चे जो उसके साथ मैंने रहने दिए हैं, उसको मैं गाइड करते करते वो एरिया में ले जाने का प्रयत्न करता हूं। उससे क्या होता है कि आराम से वो बच्चों का कॉल आता है बास्केट में से, तो अट्रैक्ट होता है पेरेंट्स बर्ड और वहां पर जाके वो इजीली अंदर चला जाता है। यू कैन नाउ कैच इट एंड उसको हम अच्छी जगह से राइस फील्ड या फिर फ्लडेड फील्ड या नियर बाय कोई भी वाटर बॉडी है, उसमें हम उसको रिलीज कर सकते हैं। कई बार ऐसा मेरे पास ऐसा इंसिडेंस हुआ कि आसपास के जो एनजीओस है, विद्यानगर नेचर क्लब है या फिर एक फाउंडेशन है, वहां से कई बार ऑर्फन बर्ड ये डकलिंग्स मिल जाते हैं, क्योंकि मालूम नहीं है किसी को कि ये डकलिंग्स ऐसे ही नहीं है, उसका पेरेंट्स आसपास में ही होगा। मगर ये हमारा अभी इंफॉर्मेशन, अभी ये जो सोशल मीडिया है, उससे मुझे बहुत मजा आता है, क्योंकि मैं अपना ये इंसिडेंट शेयर कर सकता हूँ। शेयर करने से फायदा ये होता है कि जिसको मालूम नहीं है कि कैसे इसको हम प्लेस कर सकते हैं, कैसे हम उसको इजीली साथ में गैदर करके उसको नियर बाय वाटर बॉडी में ले जा सकते हैं। तब तक ये ऑर्फन हो जाते हैं बच्चे अपने आप, या तो फिर कुत्तों या बिल्ली का शिकार हो जाते हैं। बर्ड जो एडल्ट बर्ड है वो फ्लाई करके उड़ जाते हैं, तो उसको हम बचा नहीं सकते हैं। मैं कई बार ऐसे ऑर्फन बर्ड आते हैं, तो मेरा बर्ड वॉचिंग का जो हॉबी है, उससे मैं कंटिन्यूअस, मेरा ऑब्जर्वेशन तो रहता ही है कि कहाँ पर बिस्लिंग डक के बच्चे के साथ पेरेंट मूव कर रहे हैं वाटर बॉडी में। तो मेरे पास जब भी ऐसे बच्चे आते हैं, तो मैं, अगर वो मुझको ऐसे पेरेंट मिल गए जिसके पास बच्चे हैं, तो मैं उसके साथ जो ऑर्फन बच्चे हैं उसको मैं प्लेस कर देता हूं, या तो फिर थोड़े 2-5 दिन मेरे साथ घर में ही उसको जो पोल्ट्री फीड है, जो ग्रोवर मैश बोलते हैं, उससे फीड करके उसको मैं थोड़ा रेज करने का प्रयत्न करता हूं। बाद में जाके जब भी मुझे ऐसी व्हिस्लिंग डक की पैर मिल जाए या तो रेस्क्यू आ जाए मेरे पास, तो उसके साथ वो बच्चे को मिक्स करके उसको रिलीज करता हूं। उसमें कई बार ये जो रेस्क्यू करने में ध्यान ये रखना है हमको कि ये बच्चे को जब भी खा ले, हम थोड़ी सैलो डिश में उसका ये फीड, ये क्रश्ड राइस या फिर मुमरा बोलते हैं, पोहा मुमरा, उसको क्रश करके, बाजरी उसको क्रश करके, या तो फिर पोल्ट्री फीड वो पानी में देते हैं। पानी के अंदर मैं डाल देता हूं और थोड़ा ही पानी उसमें भरता हूं, जिससे वो खाने लगते हैं, उसमें से कलेक्ट करने लगते हैं फूड को। और इजीली वो उसके ऊपर आराम से वो खाने के लिए खाने लगते हैं। मगर खाने के बाद उसको हम ऑलवेज क्लीन करके पानी में रख देंगे तो अपने आप क्लीन हो जाएंगे। बाद में उसको एक बल्ब के नीचे वार्म करने के लिए हम छोड़ देते हैं, जिससे बर्ड की जो सर्वाइवल रेट है ना वो बहुत बढ़ जाता है। जो मैंने धीरे धीरे सीखा है। कई बार ऐसा होता था, पहले मुझे ये टेक्निक नहीं आती थी, मगर आप जो टाक साहब है और उनसे ही मैं सीखा हूं ये सब, कि थोड़ा प्यार, थोड़ी टेक्निक और टेक्नोलॉजी और साइंस सब मिला के जब करते हैं तो ये सब इजीली हो जाता है। जब भी रेस्क्यू में बर्ड आता है लेसर विस्लिंग डक, तो मैं बास्केट में बर्ड के बच्चे, उसकी पेरेंट्स, लेसर विस्लिंग डक ऑलवेज इन अ पैर में ही होते हैं, वो मेल फीमेल साथ में ही रियर करते हैं। दूसरा जो हमारा एक डक है वो है नॉब बिल डक, वो सिर्फ फीमेल ही रेस करती है बच्चे को। मगर ये जो विस्लिंग डक है वो इजीली आपको वाटर बॉडीज में बच्चों के साथ अगर आप आराम से ऑब्जर्वेशन करेंगे तो ये जो रेनी सीजन है उसमें मिल जाएंगे। तो कई बार ये जो डकलिंग्स है, डकलिंग्स को रेज करना बहुत ही मुश्किल काम होता है, मगर वो आसान तब हो जाता है कि हम उसकी टेक्निक सीख ले। मेन उसको बॉक्स में रखना, हर रोज उसको 2 या 3 या 4 बार उसको फीड करना, फीडिंग के बाद उसको क्लीन वाटर में रख के, वो अगर क्लीन हो जाए बाद में अच्छे से बल्ब के नीचे रख के उसको वार्म देना, आराम से बच्चे रेज हो जाते हैं। ये आप देख सकते हैं कि जो मैंने ये बर्ड रेस्क्यू किए थे उसको रिलीज किया था, बाद में धीरे धीरे वही वाटर बॉडी में पेरेंट के साथ रेज हुए और अच्छे से बड़े हो गए। दूसरा जो एक इंसिडेंस है वो नॉबबिल डक का है। नॉबबिल डक भी एक रेसिडेंट स्पीसीज है और मेनली जो बड़े बड़े पेड़ है जिसकी कैविटी में ये नेस्ट करता है, वो कम हो जाने के बाद हमारे विलेजेस के जो बड़े हाउसेस की जो कैविटीज मिलती है उसको, उसमें ये बर्ड नेस्ट करता है और मोर देन 20-25 एग ले करता है। मैंने जब ये स्टार्ट किया था आज से 22-23 साल पहले, 22-23 साल पहले मेरे पास एक मेरा फ्रेंड लेके आया एक बकेट में 24 डकलिंग्स। मुझे भी तब मालूम नहीं था कि ये इसका हैबिट कैसा होता है, वो कैसे बिहेव करता है, क्या होता है। तो मेरे पास बर्ड आ गया, मैंने ढूंढने का बहुत ट्राई किया, मगर मेरी एग्जैक्टली साइट नहीं थी कि कहाँ पर मुझे ये पैर मिलेगी। तो मैं खेतों में बहुत घूमा, उसका ये बच्चों का कॉल से अट्रैक्ट करने का बहुत ट्राई किया, मगर नहीं मिला। तब जाके मैंने उसको रेज किए थे, मगर वो जब रेज किए तब मुझे मोर देन 3.5 मंथ लगा था ये रेज करने में। और बाद में मैंने उसको रिलीज किया था वाइल्ड में, वो बाद में बताता हूं, मगर इसकी टेक्निक मैं बता देता हूं कि नॉबबिल डक जब भी आपको मिले, आपके अभी तो सिटी में, बड़े बड़े सिटी में भी ऐसे नॉबबिल डक ने स्टार्ट कर दिया है नेस्टिंग करना। तो उसका जो जब भी वो रेस्क्यू में आते हैं तब नॉबबिल डक बिल्कुल बीच सिटी में आ जाते हैं। तो बीच सिटी में आने के बाद करना क्या है, तो मेन उसके जो बच्चे हैं उसको हाफ या 25 बच्चे हैं तो 20 बच्चे को आप एक अच्छे से बकेट में, बास्केट में या बकेट में उसको भर लो। उसके जो बच्चे का कॉल है उससे पैर ऑलवेज अट्रैक्ट होता है और फीमेल थोड़ा प्रयत्न करने से उसके पास आ जाती है। तो तभी उसके साथ 2-5 बच्चे उसके साथ छोड़ देने का, वो छोड़ते हैं तो बिकॉज ऑफ दैट डकलिंग्स वो वहां पर रह जाती है। उसको थोड़ा प्रोटेक्शन देना इंपॉर्टेंट है, क्योंकि वो प्रोटेक्शन देंगे तो ही वो फ्लाई नहीं करेगी, नहीं तो वो फ्लाई करके चले जाएगी। बाद में क्या करना है, तो वही या तो मॉस्किटो नेट जो हम रात को डबल बेल के लिए यूज करते हैं वो, या तो फिर हेलो
भट्ट साहब,
हाँ
15 मिनट्स प्लीज।
ओके ओके। तो अगर मिल जाए तो उसमें हम जैसे विस्लिंग डक को भी हम गाइड करके एक रूम में ले जा सकते हैं, वैसे ही इसको भी हम ले जाके उसको वहां से कैच करके डकलिंग्स और डक को राइस फील्ड या फिर बड़ा पॉन्ड हो उसमें हम रिलीज कर सकते हैं। ये मैंने जो रियर ये बच्चे रेस कर किए थे उसको मैंने ट्रेन किया था वाइल्ड में कैसे सरवाइव करें। तो उसको मैं राइस फील्ड के जो छोटा ग्रास होता था वो मैं देता था, वॉर्म देता था, उसको एनवायरनमेंट देता था, और बाद में जाके उसको मैंने राइस फील्ड में रिलीज किया था। सेमली सिटी में जो अर्बन आउल है वो बान आउल है, वो बान आउल का भी मैंने एक बार मेरे पास 5 बच्चे आए थे, उसकी मदर भी एक्चुअली पोचर के पास आई थी, तो पोचर के पास से छुड़वा के एनजीओ मेरे पास ले आए थे। फर्स्ट टाइम मेरा उसका एक्सपीरियंस था, मगर उसको मैंने ऐसे रेस किया था कि बाद में मेनली जो बच्चे जो थे वो जुवेनाइल हुए, बाद में उसको नेचुरल एटमॉस्फेयर में अगेन वाइल्ड में चला जाए, इसलिए बच्चों को ट्रेन किया था। और वो जो बच्चों को मैंने ट्रेन किया था, आप देख सकते हैं यहाँ पर कि एक बच्चा जो है वो एक मैंने जो बॉक्स बनाया था जिसको कवर किया था कि आसपास से कोई बर्ड उसको परेशान ना करे, तो उसमें वो रहता था कंप्लीटली। नाइट को वो हंटिंग के लिए जाता था और फिर आके उसमें रहा था। एक्चुअली वो अप्रोक्सीमेट 1 ईयर तक उसमें रहा, बाद में उसने चेंज कर दिया था। कई बार बड़े बर्ड जो एंडेंजर स्पीसीज है वो कई बार इंजर्ड होके आते हैं, तो मेरे पास थोड़ा नॉलेज था इसका, इसलिए हमने एक्सरे लिया था। डॉक्टर स्नेहल पटेल उसने उसका प्रोसेसेस बनाया और फिर बाद में इंद्रोडा पार्क में हमने उसको रिलोकेट किया। ये जो बर्ड्स का जो एंडेंजर स्पीसीज है उसके लिए अभी गवर्नमेंट भी बहुत अच्छा कुछ काम कर रही है। गवर्नमेंट भी उसके लिए रिहेबिलिटेट करने के लिए उसका बहुत प्रयत्न कर दी है। तो वेटरनरी साइंस के स्टूडेंट्स के पास ये बहुत बड़ा फील्ड है कि वो ऐसे जो रेयर बर्ड है, ट्रिकी बर्ड्स है, उसको भी आराम से और इजीली उसको रेस करके फिर उसको वाइल्ड में रिलीज कर सकते हैं। ये जो आपको मैंने बताया था येलो फुटेड ग्रीन पिजन जिसको मैंने रेस किया था, वो 2 बर्ड थे। एक बच्चा था, उसको मैंने 3 साल रखा था, मगर उसको प्योरली मैंने वाइल्ड बेरीज के ऊपर रखा था। और दूसरा जो था वो इंजर्ड होके आया था मेरे पास, मगर थोड़ा 2-3 दिन का ही उसको रेस्ट चाहिए था। वो रेस्ट के बाद वो अगेन वाइल्ड में चला गया। वैसे कई बर्ड ऐसे आते हैं जैसे ये कॉलर डव है, ऑर्फन मिल जाते हैं। एक बहुत बड़ा इंसिडेंस बताता हूं कि आपने सबने चातक पाइड क्रेस्टेड कुकू का नाम तो सुना ही होगा। वो यहाँ पर आता है और जैसे कौवे के नेस्ट में कॉयल एग ले करती है, वैसे ही चातक बैबलर के नेस्ट में एग ले करता है। तो कई बार ऐसे ट्रिकी बर्ड आ जाते हैं। अभी चातक का बच्चा मेरे पास रेस्क्यू के लिए आया, मीन्स ऑर्फन मिल गया किसी को, तो मैंने मेरा सेंस लगा के ऐसा सोचा कि उसको मैं कोई एक ये जो वाइल्ड जो बैबलर है, जंगल बैबलर, तो उसका बच्चे के साथ मैं उसको प्लेस कर दूं। तो ये करने के बाद इजीली वो प्लेस हुआ था। कई बार एक दूसरी टेक्निक मैंने जो की है और उससे बहुत अच्छा फायदा मिला है, वो आपको बताता हूं कि जो भी बर्ड है उसके अगर नेस्टलिंग मिल गए आपको, तो वो दूसरा बर्ड के नेस्ट में प्लेस करने से इजीली वो एक्सेप्टेबल हो जाते हैं। जो हमारी मान्यता है कि वो उसको स्वीकारेगा कि नहीं, वो ऐसा नहीं होता है। ये बुलबुल जो है, ये बुलबुल ने अपने 3 बच्चे और मैंने एक 4 बच्चों वाला नेस्ट जो एक पेड़ की डाल गिर गई थी उसके साथ नीचे आ गया था और दूसरे गांव से मेरे पास आया था, उसको मैंने उसके साथ प्लेस कर दिया था वो नेस्ट को, और बाद में वो इजीली उसने 7 बच्चे को रेज किया था। तो इट इज अ टेक्निक, समवॉट टेक्निक यू कैन डेवलप एंड एडप्ट, तो ये हो सकता है। ये कॉमन आयोरा है। कॉमन आयोरा बहुत स्मॉल एंड डेलिकेट बर्ड है। उसका बच्चा मेरे पास कहीं से आ गया था, तो मैंने उसके लिए क्या किया, मेरा एक ही मकसद होता है कि मैं ढूंढता हूँ कि कहाँ पर उसके पेरेंट्स मिल जाए, कहाँ से ये बच्चा मिला, कहाँ से ये बर्ड मिला, तो उसी प्लेस पर मैं उसको ले जाके वहां ही रिलीज करना मैं मुनासिब मानता हूँ। उसका कारण यही है कि जो ये बच्चा है, तो बच्चे को मैं रेज करने जाऊंगा ना, तो वो एक तो उसका फूड है इंसेक्ट्स वगैरह, तो इतना इंसेक्ट्स लाना मेरे लिए डिफिकल्ट हो जाएगा, और फिर उसको मैं वाइल्ड में प्लेस भी नहीं कर सकूंगा। तो मैं ये टेक्निक करता हूँ कि उसका जो बच्चा जहां से मिलता है, वो बच्चे को मैं उसके पेरेंट्स ढूंढने का प्रयत्न करता हूँ, और पेरेंट्स के पास ले जाके उसको रिलीज करता हूँ। तो उसमें बहुत हमको फायदा मिलता है। मेरा जो बिलीफ है वो रेवरेंस फॉर ऑल क्रिएशन। हमारे आसपास के जो भी पंछी है, जो मैमल्स है, जो फॉरेस्ट के बर्ड है, फॉरेस्ट के मैमल है, और उसको कंजर्वेशन के लिए जो चाहिए वो वनस्पति मीन्स पेड़ है, या फिर आसपास के फ्रूट बेरिंग, फ्लावर बेरिंग जो ट्रीज है, उसको हम बोए, ज्यादा उसको उगाए और उसको एनवायरमेंट दे, तो जाके हम ये कंजर्वेशन, ये रेस्क्यू भी करेंगे, रिहेबिलिटेशन भी करेंगे, मगर उसका एटमॉस्फेयर उसको नहीं मिलेगा तो वो बचने का चांसेस बहुत कम हो जाएगा। तो जब भी मैं ये काम करता हूं मेरी लाइफ में, तो मुझे जैसे मैं कुदरत की गोद में बैठा हूं ऐसा लगता है, इसलिए ही ये सब मैंने काम इतने सालों से करता आया हूं। और माइनर एफर्ट्स जो मैंने डाले हैं, वो माइनर एफर्ट्स से इतना बड़ा रिजल्ट्स मिला है। ट्रिकी बर्ड्स को रियर किया है, उसको रेज किया है, ट्रिकी बर्ड्स को वाइल्ड में एस्टैब्लिश किया है। तो मेन जो कंसर्न है ना कि ह्यूमन के पास जब आ जाते हैं तो वो वाइल्ड में नहीं जा सकते, बहुत टफ हो जाता है। मगर थोड़े एफर्ट्स डालेंगे उसके लिए, थोड़े हमारे जो बेसिक जो प्रिंसिपल है, उसको फूड जो देना है, उसका ध्यान रखना है, उसको एटमॉस्फेयर देना है, वो अगर हम नेचुरल देंगे तो आराम से वो एस्टैब्लिश हो जाएगा। सो थैंक यू वेरी मच टू वेटरनरी साइंस कॉलेज, डॉक्टर टाक साहब, डॉक्टर अलावत साहब, Dr. Rag Saib, and all students for giving me such an opportunity to share my all.
Hello.
Hello.
What, sir?
Yes, yes.
What, sir? Nicely you have presented your lecture. I also welcome our respected principal and dean, Dr. Taksar is here.
Yeah.
Dear student, do you have any question? So, yes, sir.
Hello, Yagnesh, કેમ છો?
કેમ છો સાહેબ?
બસ મજામાં. રામ રામ.
રામ રામ. મને ઇન્વાઇટ કરવા બદલ ખૂબ આભાર. સાહેબે જ મને બહુ ઘણું બધું શીખવાડ્યું છે. પ્રેમ કરવાનું બહુ શીખવાડ્યું છે. સાહેબ દર વખતે મને એક વસ્તુ કહે, સાહેબ એટલે સાહેબને જો સ્ટુડન્ટ્સ હોય ને, સ્ટુડન્ટ્સ ઘણી વખત સમજી ના શકે, પણ સ્ટુડન્ટ્સને સાહેબની ક્વોલિટી કહેવી પડે તો પછી સ્ટુડન્ટ એ ક્વોલિટીનો ઉપયોગ કરી શકે. સાહેબ ઓલવેઝ, હું જ્યારે બી કંઈ પણ મેડિસિન માટે અથવા તો ટ્રીટમેન્ટ માટે પૂછું ને, તો ટ્રીટમેન્ટ તો કે, પણ સાથે એક વસ્તુ જરૂર કે થોડોક પ્રેમ તમારો છે ને એ ઉમેરશો એટલે બધું થઈ જશે. અને ખરેખર થઈ જતું હતું. એટલે આ જ મહત્વ છે કે આવા જો પ્રિન્સિપાલ, આવા ડીન મળે તો સ્ટુડન્ટ્સ ઘણું બધું મળશે. મને તો નતું મળ્યું, પણ મને આવા સાહેબો ઘણા બધા મળ્યા કે જેમણે મારી જિંદગીની અંદર બહુ બધું નવી ટેકનિક્સ, નવું જાણવા મળ્યું, નવી ટ્રીટમેન્ટ હું શીખ્યો. તો એને લીધે ઘણા કામ થઈ શકે. અને આવું જો આવી થોડીક મનની એવી મરજી ડેવલપ થશે તો જરૂરથી વેટરનરી સાયન્સના જે સ્ટુડન્ટ છે એમને એટલી બધી ઓપોર્ચ્યુનિટી મળશે અને એટલું બધું શીખવા મળશે, એટલું બધું કામ કરવા મળશે. ને એ કામ કર્યા પછીનો જે આનંદ મળશે ને એ અદ્ભુત છે. જે પણ ટાક સાહેબે જ મને કીધું છે, એ ખુશ થાય ને અને એની અંદરથી આપણને આનંદ મળે, એ જ્યારે રિલીઝ થાય વાઇલ્ડમાં અને ફરી પાછું એના નેચરલ હેબિટેટની અંદર, નેચરલ પોપ્યુલેશનની અંદર જ્યારે ફરી પાછું જાય, ત્યારે જે આપણને આનંદ મળે એ કંઈક જુદો જ આનંદ.
યજ્ઞેશ, તમે પણ ધર્મજનું વિહંગ છોડી અને અમદાવાદના ચબુતરામાં ગયા છો?
ના, એટલે એકચ્યુઅલી એવું છે કે થોડાક કામોને લીધે થોડાક મારો તો પ્રયત્ન એક જ છે કે કુદરત સાથે જ કાયમ રહેવું. અને અહીંયા પણ એવું જ છે કે મારી આસપાસની અંદર પેલું લો ઓફ એટ્રેક્શન નથી કહેતા, જ્યાં તમે જાઓ ત્યાં આગળ મળી જાય. તો મારી આસપાસ એટલા બધા મોર આવે છે, મારી આસપાસ બાર્બેટ, વુડપેકર બધા આવતા થઈ ગયા છે. મને બી મજા આવે છે અને એટમોસ્ફિયર સરસ છે આજુબાજુનું, એટલે મજા આવે છે.
ધર્મ જેવું સ્વર્ગ પૃથ્વી ઉપર છે નહીં બીજું.
સાચી વાત છે. ત્યાં જતો આવતો રહું છું.
ધર્મ જ યાદ આવે છે.
અરે સવાલ જ નથી. એ તો તમે જ્યારે યાદ કરશો એટલે હું ત્યાં હાજર રહીશ.
Our Gir forest and Girnar around is equally comparable to Dharmaj, but it has its ecosystem and biodiversity is so rich, not only of the birds, but the animals, human, even the plants and immortal like mountains, rivers, geographical and all landscaping naturally so diversified. Everything is available in around Gir. So, I really miss Dharmaj, but Gir has given a lot for the like-minded people, especially the basement of the Gir, around more than 50 kilometers area by its dayas. So it is very biodiversified for people.
Really, and surround also much beautiful. There is a sea nearby you, forest, mountain, everything.
So we invite your ornithology society, Pakshi Premione, to visit once the basement of the Girnar, Tadeti, very popularly called as Tadeti. you will find lot of variety of the birds, plants, even the unique creature of the god, that is Asiatic lion, is also equally roaring sometime in our campus also.
Yeah.
So please.
સાહેબે એટમોસ્ફિયર ધીરે ધીરે બનાવ્યું હશે ને એટલે પછી હવે એને આવવાનું મન તો થાય.
But we also expect you to visit once.
Sure.
Dr.
I will come, and I not visit, I stay also there for some learning and some.
Most welcome.
Yeah.
Most welcome.
Thank you.
I
sir.
hope rest will find there. And thank you for appearing virtually and enriching the environment.
Thank you, sir.
Keep coming and smiling.
Sure. 101%.
Okay. Thank you, sir.
Thank you, sir. Thank you.
Students query from student side. So anyway, Sri Jagdish Bhai, thank you very much for saying good words for our principal and team, sir. And thank you very much for your nice presentation. And I think that our students, they will be the bird lover or animal lover. So thank you very much, sir. Thank you very much for joining.
Thank you.
Hello, dear students. At 3 o'clock, again, we will meet for remaining lectures. So thank you very much for joining.
I have also attended your conference organized by you, sir.
Acha, you attended that. Acha, very good.
Yeah.
I'm Dr. Savarkar, sir.
Yes.
Professor in veterinary extension.
Okay, sir. Namaste.
Dr. Sharma is there. He is working as a professor and head.
Namaste to all, sir.
Dr. Vada is here. He is working as a associate professor and head in Department of Gynecology.
Acha. You are a very good team now organizing such event.
What about your university, sir? I think that new university has created.
University will be created shortly. Now, already government has declared in the assembly, and only is allocation of fund. They have only allocated 20 crores for this year.
Okay.
And there is a proposal of 800 crores submitted yesterday by a committee. That is to be approved by the finance.
Any gazette notification is issued?
The notification means only in the budget it is declared. Otherwise, cabinet decision and all they will have a press meet and they will declare.
Okay.
So in our Gujarat also, the university was declared in Kamden as a Kamden University.
Yeah.
Since last six months, still our college is not integrated to that university. Still we are in the different state agriculture university.
Acha.
Sir, you can check your PPT.
Should I share now?
No. Please check your PPT.
Only by sharing, na?
Yeah, just share and then minimize, sorry.
Acha. You have made me the host.
Yes, sir.
कितने लड़के आ गए अपना नीरज भाई?
Is it displayed?
Yes, sir. Yes.
हाँ कोई दिक्कत
Sir, just like your area, sir, our area is also famous for Asiatic lions. Sir. We have also very good diversity, biodiversity, especially wildlife.
Oh, acha.
We are also routinely conducted the postmortem of the number of the wildlife species, especially like leopard. But we don't have elephant just like you, sir.
Yeah.
We don't have elephant, sir.
Acha. But now elephant is also important for the country. As you told about the carnivores, lions or tiger, that is also important. At the same time, also wild ruminants are now a problem, specifically as far as the disease is concerned.
Yes. University is also having IDP project, no?
The trainees, the participants are mostly from your place or from others outside?
From our place only, sir.
From your state?
The university is also having this IDP project.
Sir, now I want to know the participants are only from university or from outside?
Sir, myself, Dr. A.K. Sharma.
Yes, sir.
Sir, I'm asking you that you are also
actually all your face are little bit smaller in size, so I am unable to see the face fully. Hello.
Sir, I am asking that your university is also having this IDP project.
Yes, we are having IDP project.
So under this project, have you completed your international training part?
International faculty training. They have selected from last year, but it is not implemented. Only the students have come. They have come before Corona. In the meantime, no teachers are deployed. They were selected, but it is not implemented till now.
The same case with our university, sir.
Yes.
In our university also have selected some of the faculty, but could not send for training in outside the country because of the Corona. And still, this challenge is continue. So it is very difficult to send.
Yes.
Out of the two faculty is over here.
No.
Dr. Fefa has already been selected.
Sir, I have been selected for the foreign training under the guidance of Dr. Meti Kupal. I think we have met at Mizoram as well. Meti Kupal, the mast cell tumor specialist.
Yes.
And second faculty is Dr. Tangadia.
I know him.
I think you know him.
He came here to Odisha also as examiner, and I usually come across with him at many occasions. Is he there?
Hello, sir. Good afternoon.
Great. Dr. Trangadia. Yes, I am not able to see the face clearly because you all are sitting. No, not that. Because you all are sitting in a room in a meeting board. That's why face are very, very small. Your life size. No problem. Now I am able to locate you. Yes, your body language will say where is Dr. Trangadia.
Yeah.
So how is that?
Yes.
So you are also going to organize one seminar, no?
Yes, one webinar tomorrow. Tomorrow there will be one webinar. Again on Saturday also there will be one webinar. Dr. M.K. Gupta will come. That post flyer is not yet circulated.
Yeah.
Is it your department or it is your university or college?
Pardon, sir?
Is it the place where you are sitting? It is a departmental conference hall or something? Where are you sitting?
No, sir. So we are seated at Veterinary College conference room.
Conference hall, yes.
Dean officer. It is a within a district. Yes.
Acha. Ha.
Sir, very good afternoon.
Yes, sir. Afternoon. Good afternoon.
Good afternoon, dear students, trainees and friends. So at morning hours, we have completed 3 lectures, and you got a very good information from these eminent speakers. In afternoon session, we have also very good speakers, and regarding this present topic that is to be delivered on challenges and opportunities in wildlife disease diagnosis. You know, these students, wildlife disease diagnosis is a great challenge, and nowadays there is advanced technology is there. It is very easy now to detect various diseases of the wildlife, and these are the opportunities for the veterinarians for making their career in future also. So we are having a very good eminent scientist and academician, Dr. S.K. Panda, Professor and Head, Department of Veterinary Pathology, Orissa University of Agriculture and Technology, Bhubaneswar. So again, I welcome you, sir, and also I welcome all the participant trainees and friends. So now, before the start of this lecture, I would like to request my colleague, Dr. Phafer, to introduce this eminent speaker.
Thank you, sir. Once again, welcome you, sir, Panda sir, and the dear friends. Before going to start the lecture, I would like to say something regarding Dr. Panda sir. Dr. Panda sir, working as a professor and head in the Department of Veterinary Pathology at Odisha University of Agriculture and Technology, Bhubaneswar. He is involved in teaching, research, as well as the extension activity since last 25 years. He made more than 10 national and as well as the state awards, including the Dr. Ganti Sastri Award, the Sri Ramlal Agrawal National Award, Best Teacher Award in the veterinary faculty, and then the Best Wildlife Pathologist. He is very known personality in the pathology as a Best Wildlife Pathologist. He also diplomat in the veterinary pathology by the Indian College of the Veterinary Pathology. So during his professional career, he has guided the more than 30 PG students and 8 PhD students. He has published the 150 national and international research articles, 50 popular articles, and 2 practical manuals, as well as the 2 book chapters. So Dr. Panda sir has participated in a number of the extension activity, including the radio and TV talks. He is working act as a reviewer as well as the editor of the national and international journal. And he also organized the National Veterinary Pathology Congress 2013 very successfully at Bhubaneswar. And he has also conducted a number of the training, especially on the diagnosis and the control of the bird flu, as well as the postmortem diagnosis of the disease by postmortem examination for the government veterinarians and as well as the academicians. So with this brief introduction, now I request Panda sir to give the some information regarding the challenges and opportunities in wildlife disease diagnosis. Sir, please. Sir.
Thank you all for inviting me for such a program. We all are happy in one way that we are able to meet each other virtually for the sake of science. At the same time, we are also able to interact each other. Whenever a topic like challenges and opportunity in wildlife disease diagnosis comes to all of us, we know they are maintained in a 2 important and a different environment. One is in a pre-living condition, another also in captivity. Both are challenging. There is threat of the habitat. There is threat in disease diagnosis. There is threat in confirmation of a disease. There is threat of the veterinarian who is exposed with the wildlife diseases. Many pathogens are having very high genetic significance. We all know there is a threat to wildlife population habitat. There is deforestation, loss and fragmentation of habitat. There is human-animal conflict, establishment of increased number of protected areas and captive facilities, atmospheric and water pollution, various forms of stress, and there is close contact between wildlife, domestic animals, and human beings. So such a situation has created a very dangerous environment, more specifically the wildlife environment or wildlife habitat. By that, we are going for a very Herculean task of disease diagnosis, because of these stress, not only in the free living conditions, even also in the captivity also there is a challenge. You cannot believe, in captivity now in everywhere in the country, all the deer parks are overpopulated. There is inbreeding, there is stress, there is ill effects of the inbreeding. So there is a question in biosecurity in zoos and sanctuaries. So whether it is a free living condition or a captivity, there is all problems, challenges are there in the habitats, which is under not the control of anybody else. Either it is we can control at our level, or even the forest officials can control that. Such a situation has come now for the whole country, how to take care of the habitat. Besides, wild animals are under serious threat of diseases, and more important challenges are for us, for a veterinarian, how to diagnose a disease. But before coming to a conclusion, before coming to a suggestion of any disease, as a veterinarian, as a clinician, there are a lot of challenges. You will be surprised to know that all the wild animals, specifically in the free living conditions, are not protected against any disease. They are not vaccinated. Their domestic counterparts surrounding them are also not vaccinated sincerely. So wild animals, wildlife populations are not protected against any diseases through vaccination. Domestic counterparts are not sincerely vaccinated. So they are specifically for which the wildlife species are at risk of any type of diseases, as we all know that there is a chance of close contact of domestic animals and wild animals. Recently, I visited a place in Odisha that is a sanctuary 500 kilometers from Bhubaneswar. You will be surprised to know that 6 elephants died within a span of 1 week due to a disease like hemorrhagic septicemia, as you all know. People were asking, even also central team visited the place. Even also there was a question how it is possible that elephants are suffering from HS. People were laughing at. Then virtually, when the molecular confirmation was made, then it was thought of perhaps the disease has been transmitted from the domestic counterparts. When on the early part it was told that all the domestic populations are vaccinated sincerely surrounding the zoo or sanctuary, but later on we came to know that animals are not vaccinated. And the animals which are dying due to a disease like HS or anthrax or BQ, whatever it may be, they are thrown without a post-mortem. So you imagine a situation, a disease like a very highly infectious disease like HS, a cow has died and it is thrown without a post-mortem. The environment is now infectious. So the innocent wild elephants could be able to get the infection through that. So a matter of 1 week without diagnosis made a hue and cry. And out of all 5 deaths, all 4 deaths, the animals were putrefied. So there is a limitation in detecting a dead animal in a pre-living condition. You cannot detect a sick animal. So whatever may be the wild animal, so the most important challenge for all of us that we are not able to detect the wild animal. We are not able to see if animal is suffering, if animal is sick, if animal is diseased. So what about the collection of blood examination? What's about the diagnosis? What's about the clinical investigation? Or what's about the collection of the samples? So this is a very, very important challenging situation for the diseased animal. There is a limitation in antemortem investigation. At the same time also, there is a limitation in any therapeutic approach. There is even limitation in conducting the postmortem timely. We are not getting a fresh carcass. Again, also it is an arduous task to expose a carcass if it is more than the size of a cow. Even sometimes also extension of investigation from the postmortem is also very, very difficult. Again, wildlife populations are distributed throughout the country. All the wildlife populations are handled by the veterinarians who do not have the expertise of specific discipline. Whether it is whether a microbiology is required, or a medicine is required, or a pathology is required. So a veterinarian who is having very little knowledge of all the disciplines in a specialized way is to handle the wildlife populations in a disease condition. So these are very, very difficult situations are there in the field, either in the zoo or in the sanctuaries. Still then, we are having some opportunities. If animal is suffering, at least in a captivity or in a zoo, we can take care of that. We can make a diagnosis. We can collect the blood. But again, we have to be careful whether this animal, we have to suggest, we have to decide whether this animal is to be tranquilized or not. So to simply to collect 5 ml of blood, a diseased animal is to be tranquilized. So it is again not possible. So animals are dying. Again, another peculiar thing I am telling, you will be surprised. You all know that animal will survive up to that stage without showing any clinical signs. They hide many clinical signs and finally the animal died suddenly. I have seen many requisitions that animal has died in the last night without showing any clinical signs. It is a very important thing you will get as a requisition. So you never get any clues. The caretaker do not observe properly. So critical observation of the sick animal is also not proper. Sometimes not possible. A wild animal hide the clinical signs. So with all these difficulty, we are unable to do so. The animals are dying. So even a single animal dies, that gives the best specimen for the diagnosis. That is the only way of diagnosis in wildlife. Many times people say that this is the beginning in a diagnosis because we could not do anything antemortem. Even sometimes it is a beginning, sometimes it is an end also. So postmortem findings of different organs or tissues are correlated with the possible systemic and special diseases. If possible, we can collect the morbid samples for extension of investigation, for a molecular confirmation or pathological investigation. So that has become important nowadays. This is the best opportunity available in wildlife by exposing a carcass, correlating the pathological lesions with a particular disease, to say that this is the disease. But again, we have to satisfy many people in wildlife disease diagnosis. You have to satisfy yourself. You have to satisfy forest officials. You have to satisfy the veterinarian clinicians nearby. You have to satisfy the public. You have to satisfy the forest officials. Even also you have to satisfy the media people nowadays. So the science, the challenges are many. At the same time, expectations are more in wildlife disease diagnosis. So if there is more expectations, and then we have to start so promptly, even from postmortem or necropsy. So that's why people say that this is the preliminary investigative tool, not only for detection of cause of death, but also for intelligent and scientific understanding of the disease. People will not be happy if you say that it is HS. People will be happy even also. I was giving you the example when even we diagnosed through the metagenomic analysis that it is a case of HS. We confirmed we investigated up to that level. Still then, our chief minister also convened a meeting how to control that disease. It is not that only diagnosis. You have to think of what is the cause. Again, also you have to decide the source of etiology. What is the source of transmission? So those are very, very important nowadays. Not that only we will say, so basing on the history, basing on the clinical information, basing on the circumstantial evidence, basing on the pathological lesions, and a confirmatory diagnosis, we have to detect, we have to confirm. At the same time, we have to also say, this is the source of etiology. After 1 month of rigorous investigation of the area, we could be able to find 2 dead carcasses in the deep forest of that case. With all, when the chief minister is telling to all the forest officials, veterinary officials, that you must look after, that there should not be further death. Then with all the pressure, with all political will only, we could be able to know the source of etiology, that one dead animal, a dead cow, with HS, that was also diagnosed. Then only the disease transmission, source of etiology could be able to be established. So it is not possible, but we should think of how there should be a commendable results to be achieved, either through antemortem examination or postmortem examination. Then only our challenges of investigation will be successful. So the opportunities are there, few opportunities are there. I cannot say that there are a lot of opportunities, but few opportunities to be taken care of very seriously nowadays. As I know, as I told you, that there are a lot of clients for wildlife, starting from forest officials up to media people. And again, for the sake of wildlife, we all should work together. If you consider that which type of wildlife in your place, it is mostly wild carnivores or wild ruminants. In our place, besides that, we are having elephants, but it is not that all these things are not there. We all should think of how all type of wildlife populations are to be taken care of. So simple thing I can tell here from my experience, whatever knowledge we have gained in domestic animals are to be simply applied. When I conducted for the first time in my life an elephant, I was not knowing much about the anatomy of the elephant and what's about the pathology of the elephant. But gradually I learned. Then also, so whatever knowledge we have gained in our other animals, specifically domestic counterparts, it is to be applied. Whether it is carnivores, whether it is birds, whether it is wild ruminants or the proboscides, all are important. But if we consider, then one important species like wild ruminants, their populations are almost everywhere in the country. They have posed a threat now. Almost all types of diseases we will get in a deer park, in a zoo, in a sanctuary. So they pose a threat of inbreeding, overpopulation. We should not be happy that we are having some animals are going to be extinct. At the same time, some animals are going to be overpopulated. So the animals which are going to be overpopulated, we should not be satisfied ourselves that yes, we are having large number of populations. It is not like that. Large number of populations are a threat in a limited geographical area, in a limited space in the zoo or enclosure. If there will be overpopulation, there will be ill effects of inbreeding, there will be infighting, there will be all types of diseases, and there are so many infectious diseases are there because of this congestion, because of the overpopulation. Also the disease transmission will be more, and at the same time also control of the diseases are very, very difficult, because again I remind you that all the wildlife populations are not protected against any disease, because it is not there is no question of vaccination of those animals. So very much common with their domestic cow, because why I am emphasizing wild ruminants, because our cattle population are there nearby, either cattle population can transmit a disease to wild ruminants, which can also transmit to other animals, or a disease of wild ruminants also can come to the cattle population also, because there is a close contact now. There is a close contact. Now, cattle population are also present inside the forest in many places. So these are the 3 chambered stomach members of the families. There are 4 chambered stomach, specifically Sorbidae are very, very important. Deer having antler, Giraffidae, giraffe. Then we are also having other types of things, but we are more concerned for this Sorbidae and Bovidae. Bovidae means those are having horns. Those are having horns are Bovidae. Those are having antler are Sorbidae, deer. So horns having Bovidae are also, there are plenty of Bovidae are there throughout the country. They are spread in the form of black bucks, chousinga, nilgai, wild oxen, wild sheep and goats. The size of the wild ruminants are very significantly different. The smallest being the mouse deer, largest being the giraffe. The gaur and wild buffalo are considered as the heaviest, even up to 900 kg. So these species are spread everywhere in the country. Overpopulation, ill effects of inbreeding, mortality during neonatal stage due to pneumonia, stillbirth, debility and anemia have crippled this population. And again, what I am going to emphasize, whatever our knowledge with respect to domestic counterparts, there are also significant number of incidences are there, starting from fasciola or helminthine diseases, hydatidosis in wild ruminants. Just I am giving you a case of a fasciola. You cannot think of a fasciola in a deer park. We all know that a deer will suffer from fasciola if one intermediate host like snail is there, but these animals are not going to graze in a swampy area. Continuous death of 100 deer in a zoo nearby. There was high level meeting by mayor of the city to discuss why there are deaths. After 2, 3, 4 postmortem examination, we could be able to know that there is fasciola. They were not bothered about fasciola. They were bothered that, sir, you suggest how to control this disease. So if we'll give medicine for fasciola, again, you know that in a hot treatment of a zoo where there are 200, 300 deer populations are there, who will take the medicine, who will not take the medicine? It is beyond our control. So very difficult to treat, to have a therapeutic approach for control of the disease. So gradually we could be able to ask so many things from the caretaker and all the other suppliers, how this fasciola is coming to this park. Then we could be able to know that the fodder which are supplied to this deer park, which are collected from a swampy area, which is a source of fasciolysis. So the intermediate host of snails are possible because of the collection of the swampy fodder. So on the next, after a lot of interaction, we could be able to establish the source of etiology. Then only the disease could be checked. The matter, so could be only we could be able to solve the matter after 1 month, though we could be able to diagnose the disease within a day. So this is the most important thing we have to remember, that we have to say the disease, we have to diagnose the disease with all challenges, as I told, that there is challenges in the diagnosis, and at the same time also there is a difficulty in correlating the source of the disease. And again, we have to satisfy them only if we are able to control the disease. Otherwise, there is no way to survive in wildlife, specifically for the veterinarians and the veterinary diagnosticians and the veterinary clinicians. This is the cross-section of the parasite in the liver. Parasitic diseases are highest in the country. Diseases are easy to diagnose, diseases are easy to control, but still then highest number of diseases, whether it is a mite, whether it is a blood protozoa, whether it is even fasciola, whatever it may be. It depends upon the management in the zoos. Again, the wild animals are never dewormed. They graze in a place with stagnant water, with bushes, with the population of all types of all parasites. So the animals get the infection, whether it is ecto or endoparasites. So everything is possible both in captivity and free living condition. Even amphistomes, even protozoan diseases like Babesia, Trypanosoma, Coccidia, Theileria. One case of clinical disease of theileriosis, I can show here one giraffe, Jai, 6 years male, tallest mammal. It is the tallest mammal, was transported from Alipur Zoo, Calcutta to Bhubaneswar, Nandankanan Zoo. After its transportation, within 5 days, the animal succumbed. So during the dying stage, the blood was collected. So you can imagine the investigation, it is so difficult. So blood was sent immediately after the death, but we decided that first let us conduct the postmortem. When we conducted the postmortem of this Joy, we found there is paleness of the oral mucosa, slight icteric abdominal surface below the skin, enlarged lymph nodes, hemorrhagic lymph nodes, congested lungs, epicardial hemorrhage, coronary vessels congestion, icteric liver. You can see the yellowishness of the cut surface. Even liver, even kidney is soft, friable, pale with congested patches, congested spleen. But most important characteristic pathognomonic lesion of theileriosis was abomasal punched out ulcers in the abomazum of the giraffe. So the case which was not diagnosed, urinary bladder with reddish urine, then microscopy changes like all congestion, then exudation in the alveoli, interstitial congestion in the kidney, lymph node showing hemorrhages, abomasal ulcer. So all these microscopic examination, counter checking of the gross examination could be able to suggest that this may be a case of theileriosis. After that, we could be able to examine the blood sample of that case also, by which we could be able to see the pyroplasms. Even actinic orange technique, molecular technique, 720 base pair. So people again laughed at how it is possible that a case which was transported and after 5 days the animal was suffering suddenly and animal died due to theileriosis. So with all pathognomonic lesions, with all molecular confirmation and hematological examination, we could be able to face the challenge that whether diagnosis is correct or not. Then in literature, it is very clearly 5 young and adult giraffe from different geographies of South Africa, including 2 translocated giraffes, showed sudden onset of disease and subsequently died. So within a day, 5 giraffe died. So there is also acute deaths in case of giraffe. So joy has given a message of joy to the living. Another giraffe is still there. So we could be able to know that yes, from the pathological investigation, we could be able to establish that theileriosis is still there in giraffe. So a tropical country like India, where the vectors, we all know, is a challenge, specifically the vector population, whether it is a tick, whether it is a mosquito, whether it is a fly, all the blood protozoan diseases are predominant and significant because of these high vector populations in the zoos and sanctuaries. So that to be careful. So these are very, very important challenges, not only the diagnosis. So the source of the disease, transmitting agents, habitat, those are to be taken into consideration while we diagnose a disease, because we have to control their disease, we have to suggest something. So diagnosis will lead to control. FMD is still there, has been well recognized in captive ruminants. Many zoos have already experienced, starting from Kerala up to Odisha. So you will find the lameness. We all know that. Even also in the interdigital space, no zoo in the country is free from tuberculosis. A very old, established, hard disease, as we all know that till today also tuberculosis is not controlled in human beings. What about animals? Our animals, specifically the zoo animals which are maintained in our, a single case of tuberculosis is the source of the disease in the whole herd. So the whole herd will suffer within a short period. And again, the disease cannot be controlled. As you all know that this needs a treatment where it needs a long-term treatment. The medication for more than 4 months are required for control of a disease like tuberculosis, and specifically orally. So a very important challenging situation in wildlife treatment is oral administration. So the deer population will not take consistently for 4 months. Once upon a time also we tried, we have seen tuberculosis in a deer, bear, many species. It is such a disease. It is a disease of almost all the species, and it is a disease of almost all the organs, starting from lungs up to brain. So very easy to diagnose. At the same time, very difficult to treat, though treatment is there. So a long-term medication is required, but again, isolation is difficult. Nowadays, government is thinking of have a safari. In zoos, we have suggested for safari, though there is a deer safari, there is a bear safari. So many safaris are coming up now, so that there should not be any close association of animals in the confinement, in the captivity. So that is also another important choice for control of these diseases, because you cannot give any medicine. If you give, even tuberculosis medicines are also causing upfeed. So animals will not take their feed properly. So they will die due to starvation. So how will control the tuberculosis? This is all these nodules, pulmonary tuberculosis, caseous nodules, even also in the peritoneum, in the mesentery, in the intestinal nodules, in sambar. Caseous nodules are there in the lungs, even in the granulomatous lesions. The demonstration of bacilli, acid-fast bacilli in the tissues are also very important way of diagnosis. Even PCR amplification. Anthrax, though it is very oldest common fatal specific febrile disease, the disease is also there in many animals. Many zoos have also experienced outbreaks of anthrax. Pastorella is very common nowadays. Very common. I have seen last 5, 6, 7 years, 70% of deer population which are dying due to pastorellosis. Very clear septicemic lesions like epicardial hemorrhage, hemorrhagic trachea, pneumonic lungs, pneumoenteritis. Pneumonia and enteritis will be the best to suggest a case of HS or pastorellosis in deer populations. Hemorrhagic enteritis. Through the exudates in the trachea. Nilgai, they are also dying. Subcutaneous edema at the neck region, very characteristic. The ascites, epicardial hemorrhage. I have seen several cases of pastorellosis in these wild ruminant populations. Endocardial hemorrhage. If in presence of fibrinopurulent exudate in the alveoli, demonstration of gram-negative bipolar organisms in the bacteriology laboratory would be able to establish this disease. So all the diseases which are very common in domestic counterparts are the same. They are in wildlife populations, specifically nilgai. I have seen many cases of severe muscle hemorrhage, of BQ cases, epicardial hemorrhage, coagulative necrosis of the muscles, demonstration of gram-positive bacilli, subterminal spores. So you imagine there is a BQ in a zoo or in an enclosure means it is gone. The spores will remain long. So every year just before shower we will experience that. And you cannot believe for last 3-4 years in Nandakanan Zoo we could be able to experience that. How this is happening? They are surprised, sir. We are giving treatment, everything. Even also they have started vaccination. They tried. Still then we decided that you change the top soil of the zoo because the spores will vegetate immediately after first shower. So the disease will reoccur every year just immediately after shower, first monsoon. So after that, the topsoil is removed, then only the disease could be able to control. So we have to think of etiology and also source of etiology always. So all these disinfection, and again colibacillosis is very common because you know that water infection, waterborne diseases cannot be ruled out in wildlife populations. Peritonitis, enteritis, pericarditis are very, very important pathological features related to E. coli infection to be taken care of. Several serotypes of E. coli have been isolated in different zoos because of water. And we know that we are not able to get water always for us, which is sanitized, which is safe. So it is very difficult for safe water drinking to the wild animal species, whether it is in free living condition or even in captivity. Injury is very common. As you know that antlers and the horns in fighting are very, very important possibilities of injuries. So this is a large rupture. This is a rupture by a horn injury. This is a dog bite, because once one animal is sick, that animal will be isolated, then that will be chased by the dogs in free living. That is very common. So never dogs are chasing the deer populations if the deer population are not sick. Sick deer will be isolated, will behave abnormally, and will be chased by the dogs, and they will die due to the disease associated with the dog bite. These are all dog bite cases. Capture myopathy. You cannot depopulate any deer park. Population is all right, but overpopulation is dangerous. If you try to reduce the number of population in the zoo or anywhere, then the deer will die while you will chase. The deer will die while you will tranquilize. The deer will die while you translocate. So they suffer from capture myopathy. Even they will die within an hour, or even they die after 4 weeks of capture. So the capture operations are not encouraged specifically in small ruminants, specifically in the deer populations. A translocated wild buffalo died due to dehydration. So translocation is very, very important. You see how the muscle is dehydrated, so markedly congested. So if an animal is translocated 500 kilometers without a fluid therapy, so that is happening. Dehydrated contents in the stomach, in the rumen. Animals are being fed with any feed which are not tested properly, whether this animal is a waste feed, whether this feed is a stored feed, whether this feed contains aflatoxin, nobody knows. Once animal dies, if you find cirrhosis, jaundice, icterus, this type of a cirrhosis in the liver has given even also fibrotic proliferation of the liver histologically, bile duct proliferation. So these are all indicative of a cirrhosis which are attributed due to the feeding of feed which are supplied by the supplier, and those are having aflatoxin. So the mycotoxins are also a threat nowadays. The proper feed is not supplied. Even polythenes, you will find a lot of foreign bodies. You will find a lot of foreign bodies. People are throwing with a fancy lot of foreign bodies, the polythenes. So we all know that that is a hazard, not only for deer population or wild ruminants, even also for domestic counterparts. So there will be impaction. So the deer population is at risk, elephant population is at risk, your tiger and lion population is at risk, bear populations are at risk. Elephants are also at risk. Elephant enjoy a special status in India very high level of attention and affection from its people, remained an integral part of the social, cultural, religious and economic life of its people. India accounts for over 50% of Asian elephant population. But the most important challenging situation, every day there is death of one. Every day in this country, one elephant is dying. So very precarious situation. Whether it is due to poaching or whether it is due to an infectious disease, that is to be taken care of. Expanding human population has put tremendous pressure. There is International Union for Conservation of Nature has placed Asian elephant in the endangered list. It is a scheduled one. There is a directorate of Project Elephant, as we all know, but more death cases are there due to poaching, accident, electrocution. Before that, as a veterinarian, we should know that it is having similarity to the equines. Technique for equines are for post-mortem is the same. This is a dead elephant. The elephant cannot sustain sternal recumbency for more than 20 minutes. This is a sleeping elephant, a recumbent elephant which is a lateral recumbency. This is a temporal gland, heavily discharged temporal gland, either in a must or in a physiologically or pathologically under stress. The proboscids are having tusks. Females are having tusks. A rudimentary tusk is tusk TUSH. Gallbladder is absent. Testes are present inside the abdominal cavity. So sex determination is also very difficult. And common causes are mostly electrocution, poaching, poisoning, train accident, but diseases are also there. Very, very wide variety of infectious and non-infectious diseases are there. Common bacterial diseases like anthrax, Pasteurella, TB, coli bacillus, salmonella, tetanus, BQ, enterotoxemia are reported in elephants. Viral diseases like FMD, rabies, elephant pox, herpes virus infection, blue tongue, coryza-like syndrome are also reported. Parasite diseases are also important, like nematodes, trypanosoma. Systemic affections with infectious and non-infectious etiology are very common. Infection of the GI tract, like infection of the colon, is very common. Liver diseases like hydatid cyst and fasciolosis are also there. Food diseases are very common because we know that animals which are under captivity, they remain in a very dirty place. So there will be food diseases. Foot rot, pododermatitis is much probably restricting the movement leading to death. Foreign bodies, entrapment of large objects in the back of pharynx, intestinal blockage, valvulus intersuspension and choke in esophagus can cause mortality. These are very, very important. Pneumonia, sudden death syndrome, trauma, injury, wounds. This is my first post-mortem. I was not knowing what is a stomach bug in elephant. They cause gastritis. Parasitologically, it was identified. 2 oral hooks and a posterior spiracles. Last month also we got the case. I got the report from the veterinarians, those who are doing post-mortem, they were not knowing. There is myiasis, gastritis, stomach rupture due to Cobaldia elephantis, stomach cut. Proliferation of the stomach wall, rupture of the stomach wall, the animal die. Even also many times elephants are tranquilized when they are overfed. There is a pressure from the forest officials that this animal to be tranquilized because it has caused much damage to the public. So whenever a veterinarian goes for tranquilization, animal die during sedation because of overfeeding. As you all know that animal is overfed, there is respiratory blockage, and the person who has tranquilized is blamed. So you have to be careful. You see, one elephant is tranquilized with overfeeding, with infection. You can see the stomach is overfed, stomach is impacted with the dehydrated contents. So the dehydrated contents are so much. The animal could not sustain the tranquilization, and there was a respiratory blockage. So animal sedated forever. This is the congested brain in that case. Lot of anthrax cases are there. The veterinarians are able to see the external findings like discharges, bloody discharges from all the orifices, from the mouth, from the trunk, from the anus, even also externally also there are discharges. So which could be able to say that this may be a case of anthrax, and which could be able to show the path for confirmation. Many baby elephants are captured, are rescued, and fed orally and fed manually. So as you know that a drenching pneumonia in calves, the same thing also happening in baby elephants. Many baby elephants which are rescued, they die due to aspiration pneumonia or drenching pneumonia. So now there is a message for all of us that all baby elephants to be handled so carefully that drenching should be proper, and by that at least baby elephants should not suffer from aspiration pneumonia. Though 2-3 elephants died, post-mortem examination could be able to establish the cause of death. Then afterwards, all baby elephants which are rescued are not dying due to that, because of careful administration of feed to the baby elephants. You see, there are nodular small foreign bodies are there in the lungs, aspirated foreign material, necrosis of the bronchial mucosa, foreign material, giant cells and neutrophils. This is an elephant died. This is an old elephant. So you can say that this old elephant, there is baggy pant appearance. There is baggy pant appearance. There is a temporal fossa which is very big, are very characteristic of old elephants. If an elephant is dying with this type of furrow, this type of a fossa, temporal fossa, and a baggy pant swelling of both the limbs, suspected for old age. This is an elephant diet, but you see, with this situation, which is totally putrefied, decomposed, but it was suspected that this is a rescued baby elephant which was rejected by the herd. Though it was putrefied, but the circumstantial evidence and presence of this type of a breakage of ribs could be able to establish the mutilation and trampling in the herd. This is an elephant tranquilized with transported without tranquilization. Without tranquilization, it was just for treatment of an abscess. But for better treatment, the animal was translocated to some other place. But in the meantime, abscess was healed. But this type of wound helped me to know that this animal was translocated before 1 or 2 days. And there was a fresh wound. And the animal was struggling during translocation. Though the main problem of abscess is healed, but it was a created problem of faulty translocation of the animal and animal diet. You see, peritoneal cavity is clear, but in thoracic cavity, there was blood. In thoracic cavity, there was bleeding. There was hemorrhage in the lungs. So the animal cannot sustain sternal recumbency. Due to faulty translocation without tranquilization, the sternal recumbency will cause the death of the elephant. Besides, there is infighting, there is injury, there is toxicity. This is a toxicity case. Friable soft hemorrhagic liver, hemorrhagic kidney could be able to say, could be able to suggest a disease of toxicity. Even there is also, this is an elephant with Shara bullets. There is poaching. Elephants also there is tragic death of elephants due to electrocution. You see, there are electric wire due to train accident. 7 elephants died tragically in a place in 1 day. And nowadays, the most important hot disease has spread, that is elephant endotheliotropic herpes virus infection. I have experienced 4 or 5 cases within a span of 1 week. It was very challenging to suspect a disease like EHB without a molecular confirmation. So pathological examination with the suspicion of the disease, mesenteric hemorrhage, epicardial hemorrhage, endocardial hemorrhage, even hemorrhagic gastritis. But most important is the cyanosis of the tongue, cyanosis, bluish discoloration of the tongue, and oral mucosa. Another case, same case. And so these otherwise called as acute hemorrhagic disease. So this acute hemorrhagic disease has caused death of the elephants, which was later on confirmed that it is a EHB. So for the first time, you could be able to establish and experience and report a EHB in a zoo. And nowadays, these are all the cases of EHB happening, even also in the free living conditions are also there. Of course, it is a disease of young elephants. Myocardial hemorrhage. So the elephant is a friend to man, and now indeed our turn has come to be the friend of the elephant. How these diseases are to be diagnosed? How the diseases are to be taken care of so seriously, so that at least we should, as a veterinarian, say that these are the natural deaths due to disease or due to unnatural circumstances like poaching, electrocution, or train accidents. If that we can say, then we can say that yes, this type of preventive measures can be taken care of, even if it is not possible for prevention or control of the disease. But we should know what is happening to the animal. That is more important in wildlife. In wildlife, always you cannot say that this is the disease, this is the place from where the disease has come. But always the carcasses which are dying are to be located and to be exposed for the diagnosis of the disease. And as we know that your place is mostly of a tiger and a lions, these are almost similar conditions for wild cats. Tigers are the largest of the wild cats and were historically found throughout much of Asia. And the Bengal tiger is the most numerous subspecies of tiger and is found on the Indian subcontinent. You please remember that when our zoos are overpopulated with deer, and one single tiger also can be available in a forest. They are solitary. They have large home ranges. They are umbrella species, providing space for a variety of other species to flourish. So they prevent overgrazing by limiting herbivore numbers and maintaining ecological integrity. So presence of a tiger means indicator of a healthy ecosystem. This is an apex predator. They shape the ecosystem in which they live. The presence of viable population of wild tiger is thus an indicator of the integrity, sustainability, and health of habitats they occupy. In India, the tigers are maintained for their scientific, economic, aesthetic, cultural, and ecological values to preserve for all times. India is home to more than half of the world's tiger population. Their numbers have sunk from an estimated 5,000 to 7,000 in the 1990s, when their habitat was more than twice as large. In 2006, there were 1,411, increased to 1,700. In 2014, it was 2,026. In 2018, the population has become 2,900. So it is almost 3,000 now. On the occasion of International Tiger Day, Prime Minister Narendra Modi released the results of the fourth cycle of All India Tiger Estimation. Prime Minister said that with almost 3,000 tigers, India is today among the biggest and most secure habitats. Prime Minister has expressed his happiness. There are 50 tiger reserves in India which are governed by Project Tiger. Tigers are designated as endangered, but at least India is home of a good number of tiger populations. Disease management mostly depend upon early detection. Non-domestic felids like tiger may hide signs of illness. They hide illness highest. The tiger population will not show the clinical signs always. Keepers that have daily contact with tigers are often the best persons for nothing these changes. Any change in appetite, urination, defecation, general behavior should be documented. Whenever it is a tiger or lion, urine, fecal color, quantity, consistency should be noted. Dehydration can be assessed. Other visual observation can be obtained from outside the enclosure. And antemortem investigation in tiger is also very, very difficult. Cub mortality with a large proportion of calves falling prey to infanticide and starvation. Very, very important. Adults to be the most vulnerable age class with intraspecific aggression. There is intraspecific aggression. There are wound, anorexia, constipation, diarrhea, lameness, vomiting, debility, pyrexia, alopecia, senility. So whatever systemic pathology, whatever systemic diseases we have read are possible in these species. Even canine distemper is very now. Indian Veterinary Research Institute has said that any tiger death, whatever may be the cause, but it should be monitored. It should be screened for canine distemper. This is very common and this is very important. So the disease should be monitored 100% in the country if there is death of any tiger or lions. Disease mostly parasites are one of the most significant disease of the animals like Toxopora, Capillaria, feline cytomegalosis. These are emerging fatal tick-borne diseases. Tick is a problem now. Tick are the most important nonsense host, intermediate host for almost all type of wildlife populations, even also domestic counterparts. Feline AIDS, feline panleukopenia is considered to be the most important disease in wild felines. It is thought that all members of the family Felidae are susceptible to infection with feline panleukopenia. There are reports of bacterial disease like anthrax, pasteurellosis, salmonella, E. coli, TB, Nabelil, Clostridia, Corynebacter, all are there in these species. There are record of tumor incidences. Kidney disease is with, so just I emphasize specifically lion and tigers. Those are felids and those who take mostly the beef. Kidney diseases are very common and gastritis is very common. So primary kidney or primary nephritis due to feed, due to high proteinous diet, specifically animal proteins. So they are very much prone to nephritis and gastritis. Feline in the past years, the world has witnessed the destructive strength of a viral disease like feline panleukopenia in wild felines. We have seen in lions and tigers. It is otherwise called as feline infectious enteritis or feline distemper. It is just a counterpart of parvovirus infection. I have seen anemia, dehydration, splenomegaly, gelatinous red and basophilic internuclear inclusion bodies. I will complete within 5 minutes. In India, occurrence of feline panleukopenia amongst the captive felids have been reported. Many places, many zoos have reported that. We have also reported many times, lot of animals have died. Muscles pale, liver pale, kidneys are pale, hemorrhagic gastritis, hemorrhagic enteritis. You see chocolate bloody colored contents are there in the intestine. Even also the zoo people were telling that this animal was not showing any clinical signs. After postmortem, I visited the shed. I found there were froth, vomiting materials, leftover feed, diarrhea. So you will not get always proper information, clinical information, either from the zoo people, caretaker, or even also sometimes also the veterinarian, zoo veterinarians also. So you have to see, if you conduct postmortem, if you go to investigate, you have to see the circumstance also, from where it is coming, what is the source, what are the real clinical information. You see such a hemorrhagic gastritis, so characteristic of feline panleukopenia. Liver pale, intranuclear inclusion body in the intestinal impression smears, all the histological changes, macular degeneration, desquamation of the intestine with and intranuclear inclusion body found in the intestinal epithelial cells, in the nucleus. Tissue samples of spleen, mesentery lymph nodes were also subjected for a fluorescent antibody technique. Could be able to demonstrate the feline panleukopenia viruses. So the incidence reduced after confirmation, because you know that people will say that the animals are vaccinated, sometimes it is neglected. If it is in zoo, vaccination is possible, that is also not done properly. And in free living condition, vaccination is not possible at all. In the last few years, we are getting cytogenosis, babesiosis. Very, very commonly, we are finding that weak, emaciated, anemic, jaundiced carcasses in a tiger and a lion we are getting regularly. So all jaundiced, anemic carcasses have to be taken care of. Babesiosis, even necrotic foci, bloody exudates in the liver cottages. Even you see icteric conjunctiva, ascites, hemorrhagic enteritis, bell liver. Those are very, very common. Specifically, the tick-borne diseases are coming up very seriously now in different zoos. Besides that, corneal opacity due to trips, old age posterior paralysis are also common in the tiger lion population. There is injury due to the aggression of the animal, infighting injury. Hepatic tumors, hepatocellular carcinoma is very common in old lions and tigers. We have encountered hepatocellular carcinoma many times. These are hepatic tumors, infiltration of tumor cells, and this is very important. Please remember, tiger calves are being trampled by the mother, so there is mother's rejection. So many times tiger calves are trampled by the mother and they die. So once upon a time, 5 tiger calves died due to the trampling by its mother, so that means there is a mother rejection. So there was a lot of hue and cry, and finally, now any calves, newborn calves are coming from the tigers, they are under CCTV surveillance. So if there is any aggression from the mother, immediately the zookeeper is going and disturbing the mother not to have this type of trampling deaths. Some precautions have been taken, but we should know the nature, physiology. So it is required to have knowledge on the status of the diseases in tigers, lions, elephants, even wild ruminants, both in captivity and free living conditions, which would help in regular and constant health monitoring. That is very, very important. And a disease surveillance to combat any unforeseen eventuality. That is very, very important. So people are worried now if 1 or 2 animals are dying. 1 or 2 animals are dying. But it may so happen if it is neglected, if it remains undiagnosed. So there will be some unforeseen eventuality will come, so that there will be more deaths. So people will start criticizing the diagnosis, the approach of treatment, therapeutic approach, and prevention of the disease. So we should be very careful in all the ways, so that our populations, specifically these populations, wildlife populations, should not be at risk. Though there are challenges, lot of challenges are there, we all know, but still then there are some opportunities. We have to be very careful how these opportunities are to be availed, so that the lot of clients, what I told, are to be satisfied, because a lot of people will observe us in our action of disease investigation in wildlife, in our action of treatment in wildlife, in our action of control of the disease, in our action of translation of wild animals, in our action of tranquilization of wild animals, while the animal has become very wild. So in all the action of wildlife, people will critically observe us. So we should be very prompt. We should be very sincere. We should take the help of expert hands. We should have the knowledge. The knowledge of horizon and information is very wide in wildlife. Nobody can say that I am the expert in this particular area. It is the exposure, it is the interest, it is the knowledge to be gained gradually, and we will come to a stage that yes, we can do something for the wildlife. With this, I thank the organizer for arranging such a beautiful program for the participants, and for which we all are able to discuss about wildlife, which are the most important species nowadays. People are discussing in the country and even in the world. Thank you all.
Thank you very much, sir. Actually, you have made a very nice presentation, and your photographs itself indicates your work devotion in particularly wildlife areas. Dear students, trainees and friends, is there any query? You can ask any question to speaker.
Respected sir, I am Dr. A.R. Ahlawat, organizing secretary of this event. So, a very good evening to you, sir.
Yes. Yes.
So when we were, myself and Dr. Trangadia were joking out the details of this program, like whom to invite as a speaker. When we were discussing all the probable topics and probable speakers, your name was first to pop up in the mind of Dr. Trangadia.
Yes, yes.
And he told me that Dr. Panda is a very wonderful speaker and he has experience of delivering such kind of presentations at various national and international levels. So I'm really thankful, and Pangadia has not let us down, sir. We are so much thankful to you, sir, for this presentation.
Thank you, sir.
And we will look forward to more academic engagements with you, sir.
Okay. Okay, sir. Okay, sir. So we'll close here, sir.
Sir, if there is any question, students will place it in the text box or they will email you, sir.
No problem. No problem.
Okay, okay.
Okay, sir. Thank you.
We will discuss at any time in any mode.
Sir, again, we are really thankful on behalf of organizers and college and university, sir. Thank you very much for sparing your valuable time.
Okay, okay. Thank you.
इंट्रोडक्शन हो गया yesterday
So, dear students, trainees and friends, we will move next for next lecture. That is rhino translocation as a tool for securing survival of the species. We are having eminent speaker, Dr. Kushal Sharma. Yesterday also he has delivered a very good lecture for you, and he has shared very good information regarding wildlife areas. So he is working as a professor and head Department of Veterinary Surgery and Radiology, College of Veterinary Science and Animal Husbandry, Assam Agriculture University, Guwahati. So he was awarded Padma Shri also, you know very well. So I invite Dr. Sharma and I hand over this stage to Dr. Sharma. Dr. Sharma, sir, please.
Good afternoon, sir. Can you hear me?
Most welcome.
Yes, sir. Yes, sir. Your voice is audible. And please allow me to share my screen.
Okay, sir. Just a minute, sir.
Yeah.
Allow you, sir. Now you can share your screen, sir.
Yes. So, good afternoon, all the scientists, professors, and student friends. So, after a very knowledgeable lecture delivered by my friend Dr. Panda from Odisha, now I am giving you a little bit of fresh air because he has shown most of the carcasses. So, I want to show live animals. And the animal that people from Gujarat and the western part of India have hardly seen. But I am coming to that part of my lecture when you will realize that they even existed in that part also one day. So I am talking about the veterinarian's role in conservation of these prehistoric species that existed and roamed in this part of the world. So at present we have 5 different types of rhinos, what we call extant at present, not extinct. Some varieties of rhinos have become extinct already. So we have 2 varieties of rhinos in Africa, the black rhino and white rhino. And we have 3 varieties of Asian rhinos. So many people think that the Great Indian One-Horned rhinos is the only rhino in Asia, but we also had Javan rhino and Sumatran rhino. So I am coming to the status of these rhinos, which are very precarious. Next, please. So this is the black rhino, double horn rhino from Africa. I will not go in detail, but the population is, the status is critically threatened. They have a population of 2,410 at present. Next. The next is the white rhino. Their population is supposed to be much better. The other rhino from Africa. They have 17,000 rhinos still in the wild. Next. So the difference of black rhino and white rhino. Though they are saying black rhino and white rhino, actually from color it is difficult to distinguish. The nostril part, muzzle part has a differentiation that may not be very clear from these pictures, but that is how they are distinguished. Next, please. Coming to the Javan rhino, actually the population is very, very precariously poised right now. They have only 40 individuals now available in zoos as well as in wilderness. In fact, I worked for them in Indonesia, and the odd 40 animals they are also enclosed because the protection from the poaching is very high. And the problem with this variety of rhino is that they are very shy breeder. So the breeding success is very, very poor. So that is a very bad situation with Javan rhino. So you can see in Indonesia, only in a small part, Ujung Kulon National Park, in the southern Indonesia, they are still there, of course in the enclosure. Then we have the other varieties of rhino, that is Sumatran rhino. They have a population of around 240 Sumatran rhinos, 275 precisely. They are still available. And you will be surprised to know that these 2 varieties, the Javan rhino and Sumatran rhino, which is not present in India right now, but this alongside the Great Indian One-Horned rhino, Javan rhino and Sumatran rhinos also existed in Assam and Bengal, North Bengal for that matter. So there is a document from the Maharaja Coos Behar. It's hardly 100 years old story that in one day he has killed all 3 varieties of rhino in the bank of Sonkos River, the river that is bordering Assam and West Bengal. So they existed, and from some unwritten account, we find that some of the people in Assam, like in Lakhimpur district, they use rhinos for plowing their field. They could be easily tamed, and it is speculated that they were either Javan rhino or Sumatran rhino, which are smaller in sizes. They are very good in case of muddy fields. So probably this type of rhinos were used for plowing the fields in the Brahmaputra Valley of Assam. Now, this is the Great Indian One-Horned rhino, what we have today. And the total world population is around 3,500. And the breakup I am showing, before that, I want to show the extent of the Great Indian One-Horned rhino. So, from historical account, we find that from the eastern part bordering Myanmar up to what is called Kandahar, they existed. So probably even Gujarat was also possibly at certain period they were there in the entire area. There is a historical account when Babur entered India. In one day, he killed 4 rhinos, Great Indian One-Horned rhinos in Kandahar, that is western Pakistan, where that the plane hijacked plane was taken. So that place where 4 Indian One-Horned rhinos were killed. But the various factors have caused the advancing of desiccation, the plate movement and all that has caused drying of Saraswati River, which has actually resulted in the drying of northern area, including Gujarat, because Saraswati River also has passed through Gujarat to join Arab. Now that is non-existent. Now it is called Anta Salila. Probably it's not there. So, and the whole of the northern India have become dry. Primarily, the type of forest that still exists in northeastern region, Assam inclusive, and some part of North Bengal. That kind of forest existed in whole of northern India, and unfortunately, everything is gone now. But some part, I will come to that, is bordering Nepal in Dudhwa Tiger Reserve, where we are also trying to repopulate the rhinos because historically they existed. So that is the historical map of rhino range. So I'm showing you the only viable population of rhino now exists in Kaziranga National Park in the floodplains of River Brahmaputra. Then we have some other smaller parks like Kovitara Wildlife Sanctuary, very small wildlife sanctuary of 16 square kilometers where we have about 100 rhinos. Orang National Park, again a pristine forest, small, it's only 78 square kilometers. There also we have 100 rhinos. Now the Manas National Park, it's a very big national park. That was our focus. And see, before 2008, there was not a single rhino because of anthropogenic reasons. Actually, Manas was the home for over 100 rhinos before the infamous socio-political turmoil. All the rhinos were poached by the poachers taking advantage of the fluid law and order situation. I will come to that, but also we have 270 good population of rhino in North Bengal, Jaldapara National Park. Gorumara has a small population of 27. Now I was mentioning about Dudhwa and adjoining parts, Katanya Ghat and Lagapoda. So here we have a small population, but the Dudhwa population also is a mix of some rhinos taken from Assam and 4 rhinos brought from Nepal. And here the population is slowly growing and a lot of emphasis is being given on that. And you will be surprised to know in Lal Sunara National Park in Pakistan, it is said that they also have few pairs of rhinos. Interesting. Sitwa National Park have over 400 rhinos in Nepal. Bordia 22, Suklafanta. So this complex also, now Suklafanta and Bordia are bordering our Dudhwa Tiger Reserve. So in a cluster, probably there is a prospect. And there is another prospect developing in Balmiki Tiger Reserve in northern part of Bihar, which is again bordering Nepal. So recently we have rescued a rhino. A pair of rhino is there now. The Bihar government is planning to rehabilitate that area for rhino. So that is the present situation. Now, if you see this list, you will find that the only viable breeding population appears to be there in Kaziranga. Now, why I have kept my topic as requested by you, that how translocation can actually save the habitat, save the animals. Now, it is a common sense that if you are keeping all your eggs in one basket, you may end up losing the whole of the basket of eggs. Now, see, Kaziranga is also very much threatened by the natural disasters. Like flood is a big thing. Assam is very prone to earthquake and other natural disasters. So it is not wise to keep your only viable population in one place. And then disease issue is very big because very little is known about diseases in rhino. Only little study has been carried out. So suppose everything is okay, we have protected the animal from the poachers, but we may not be able to protect them. Suppose if there is an epidemic and we will end up losing the only species, the whole species may be lost. So that is why when Manas, I'll come to Manas. So Manas was 0 and we decided that we may translocate rhinos from Manas as well as overpopulated Pavitra Wildlife Sanctuary to repopulate Manas. And then we also have chosen some other beautiful habitats within the boundary of Assam. One is Lakhua Burasapuri Wildlife Sanctuary Complex, and in the Upper Assam we have Dibru-Chokwa National Park, which is famous for the feral horses. So let us study how we have done. Now, talking about the threats of the particular species, the main threat is from poaching for the horns. You know, there has been a traditional belief, a misconception actually, a myth that the rhino horn has a lot of medicinal value. Primarily, it is thought that it has aphrodisiac value. And after a minister in Vietnam survived cancer, though he was treated with all modern anti-cancer therapy, chemotherapy, as well as surgical therapy, but some quack have given him rhino horn powder, and that unfortunately that minister survived, and that has created such a feeling among people in those oriental countries that rhino horn can cure cancer. So ultimately, there is a tremendous pressure on poaching, and you will not believe, in just 1 year, about 1,000 rhinos were killed in South Africa alone. We give a lot of protection here, but I think in 2014 we lost 36 rhinos to poaching in Kaziranga. So poaching is a big threat, but habitat loss is another. If you talk of Kaziranga, then we lost 83 square kilometers of Kaziranga to erosion of Brahmaputra. Then I mentioned about flood and erosion, disease spread and epidemics. Then lack of genetic variability, which is in the long run a bigger threat, because because they are all living in isolated islands, having no contact with other population. So the heterozygosity will be lost, and as a result, these will be doomed population. And of course, the carrying capacity. If you talk of Pavitra, you will understand that. Space constraint and carrying capacity, poaching of rhinos for its horn, and so very high poaching. You can see Oran itself 31, Kaziranga 126. Now, what strategy we are adopting for conservation of the species? We are trying to give maximum levels of protection. Activation of the information network, information sharing, or joint command like police, army, or other security forces. Then participation of local communities, because they are the beneficiaries also. Lots of tourists visit Kaziranga as well as Manas. So the local people in the neighborhood are benefited from that. So if we can involve the local communities, they are of great help, because we get information about the movement of poachers like that. Then habitat expansion, that is the other thing that we are very keen on. Like Kaziranga, we have already 6th edition, 1, 2, 3, 4, 5, 6, and so we are adding areas to Kaziranga, and that is one. Then habitat improvement. Somewhere there is the grassland is damaged because of the succession of, like the simul tree has come, or some other tree has, or woodland has dominated the grassland. So we have to manage the grassland, and because animals need the grass. And on top of everything, selection of suitable areas for translocation. And of course, we have to go for international propaganda. We are also trying to work on the government of Indonesia, government of China, as well as Thailand and Vietnam, that to create awareness among those people against the use of rhino horn. That rhino horn has no medicinal value and all that. Then talking about the maximum levels of protection, increasing the anti-poaching camps. So in Kaziranga, we have a very high density of anti-poaching camps. like one camp must see the other camp. It is like that. They are closer to each other so that we can create a situation that the poachers cannot percolate. Increasing security personnel in each and every camp, giving high level of training as well as motivation, then providing sophisticated arms. You know, in Kaziranga now our girls, they have automatic weapons. Then electronic surveillance system, electronic eye what they call, then increasing conviction rates, and here comes the role of veterinarians also, because by forensic studies and all that, if properly we can back up with forensic studies, the conviction rate of the suspected poachers will increase, and that will act as a deterrent. Then carrot and stick for good and bad works, even for girls, forest girls, if they they can protect the rhinos from poachers, they should be rewarded, and those who are showing negligence to duty will be punished. So carrot and stick. Then very important role of the veterinarians, as I have said, many animals stray out of their birds and they have to be brought back after rescue. Translocation to suitable habitat, disease investigation and disease management, assisted reproduction wherever necessary, like we are trying in Junagadh with the Laban rhino. Then captive breeding of rhino population. You know, Central Zoo Authority has 2 captive breeding zoos. One is Patna Zoo, other is Guwahati Zoo, where rhino captive breeding program is going on, and also prevention of inbreeding, where bull exchange program, that kind of programs are taken so that heterozygosity can be reintroduced. Then veterinarians are of course involved in treatment. So this particular rhino had a gunshot injury in Kaziranga National Park, and like that we have to treat and help them. Then rescue of stray ones. So this particular rhino is strayed out of Pavitra Wildlife Sanctuary in a high flood situation, and it was resting in a small riverine island in Brahmaputra called Rani Chapari, and from there it crossed the river and entered Guwahati city. So it's against all odds, we have very successfully rescued the animal, even from very high water. So that even when you operate inside the park, this particular rhino, after it was dirted, it ran into water, and from the clothing of the people you can see what was that was the height of winter in Assam. And see, some people have entered into the pond to rescue that animal. So, you know, critical situations can happen during a translocation program. These are not to be taken as a very easy task, difficult task. So, if not properly planned, can end up losing an animal. So, here also veterinarians can help. This is a horn trimming, because the rhinos will be killed for their horns. If you can trim the horn, the rhino will not be interesting for the poachers. So this is another thing. In Africa, they are doing it in great numbers, and that lady veterinarian is doing the trimming job. Then regarding habitat expansion, we must consider the carrying capacity. So as per a scientific study, one rhino needs about 1.3 square kilometer area individually. Now in Kaziranga, we have only 0.32 square kilometer per rhino. So the carrying capacity has exceeded. Now they are overpopulated. We have to think of other areas where we can shift the rhinos. Addition of areas wherever possible, then selection of suitable habitat, ensuring protection in the new probable habitat, ensuring heterozygosity by bringing new animals from different habitat. So this is the Manas National Park, pristine forest, and on the southern side of Bhutan. So this is Bhutan on the northern side, and on the southern side it is Manas National Park. And at present we have about 1,000 square kilometer area, pristine grassland area on the foothills. And on the northern side, Manas, we have a Bhutan Royal Manas National Park of Bhutan, which is about 2,200 square kilometers. So collectively, we have over 3,000 square kilometer area available. in one block. So beautiful place. But I have already mentioned that in the Boro agitation, the entire rhino population were wiped out. And so for bringing in rhino into Manas, we thought a lot of studies has to be carried out, habitat study, habitat suitability study, then the security study and all that. And after carrying out everything, only you can imagine that we can bring rhino. So that is translocation. Shifting one animal population to another population is translocation. Now, in the translocation program, the veterinarians are the primary players. So what are their roles? They have to identify the healthy animals. Well, now this is a tricky issue. Actually, as per IUCN guideline, the rhino translocation is not to be done in one go. First, you have to capture the rhino, collect all the samples for bacteriological, virological investigation. Then, until you get the report, you have to keep the animal in coma, in separate enclosure. And after you get the positive report, you cannot translocate. If you get a negative report that rhino is healthy, then only you can translocate. The reason is that we don't want to translocate diseases. But our problem is we don't have so much of resources with us that repeatedly translocating the animal. And when we put an animal in a coma, a very free-ranging animal, it will be under tremendous stress. That stress itself will cause flare-up of diseases. That is why what we have done that we have taken to ourselves to select healthy animals physically. So we think we see two things. One is the hard health monitoring, like whether any rhinos have died in that area last 2 years. And if so, from poaching or from some diseases. So negative report has to come from there. Next is physically the rhino should appear healthy. When approached, the rhino should behave normally. It should not be drowsy or it should not appear sick. Then we have to capture. And safety of the animal in all procedures after capture, that is morphometry, radio coloring, microchipping, identification marks, that is ear tagging, or notching, that is called loading in the sledging, loading in crate, transportation up to release, and even post-release management. Even after you release, the rhino may run out of the habitat. The rhino may infect and have injuries. Right now, today also in Manas National Park, one rhino has injuries. So they are calling us for treatment like that. So even after you release, you have to manage them. So now I am trying to show you the entire rhino translocation program pictorially one by one. Now this is a Veda for us, or like for every veterinarian, the person capturing or administering drugs to a wild animal simultaneously assumes the responsibility for the life of that animal. So you are responsible if you have chosen to handle that animal or immobilize that animal. Now you can see the responsibility is very, very high. Now for immobilizing rhino, you have a very narrow window for drug selection. You cannot use sedatives, doesn't work. Okay, you can only use the narcotics. So in 99% cases, we are using etorphine, the only reliable drug. In combination also, sometimes etorphine acetaminophen, sometimes etorphine with alpha-2 agonists like dilazine or miditomidine. Carfentanil is also a narcotic, can be used, but in India we could not procure it. Now, for delivery of the dirt, we need the metal dirt system, not the pneumatic dirt system. Those who are familiar with this will agree, because in the pneumatic dirt system, there is a possibility, remote possibility, maybe but there is always a possibility of drug leakage. So, leakage of a narcotic drug can be disastrous. People can die of it from poisoning. So we don't use, and moreover the rhinos have thick skin. So we prefer metal dart and the metal drug delivery system. We have two types of guns for metal drug delivery system. Metal dart, one is the capture gun, traditional, and the other is the modified disc inject gun, 60 and or 90 and model. They are very good. This is the gun, very lightweight, 2.6 kg, and even on elephant's back you can easily manipulate the gun. So these are various accessories. Primarily, this is the metal dart, and these are the various types of needle and other accessories. And once the animal, the rhino, is inside the cage, we should not use the dart system or gun system. Then we have to go for zapstick or that type of closed type of administration method. Then we have to have a checklist of other drugs, vitamin E and selenium. No, Dr. Panda you must have noticed he talked about the capture myopathy. So vitamin E and selenium combination is preventive as well as curative against myopathy. But to avoid myopathy, we should avoid chasing the animal, because animals have high glycogen, and suddenly you chase, they get nervous, and there is muscle exertion. So lactic acid can form, and lactic acidosis can damage the muscles and they enter vicious cycle, what we call as Monday morning sickness or ozosuria. It is similar. So it can be prevented. So we always keep it. Once the animal is down, immediately we do that. Tetanus toxoid, injuries are possible. Long-acting antibiotic, fly repellent antiseptic, sedatives like acetaminophen or haloperidol, azaperam, maybe dexamethasone, doxapram, the respiratory stimulant, and adrenaline. Equipment prominently marked boxes, because here we are handling narcotic drugs. So everything has to be properly labeled and very disciplined. Very, very disciplined. Disposable rubber hand gloves, water bottle, pliers, disposable syringes, blindfold and cotton plug, then ear notching equipment, microchipping and radio coloring, and basic equipment for anesthesia monitoring, stethoscope, thermometer, pulse oximeter, resuscitation unit, sphygmomanometer and cotton wool antibiotic, sample collection vial, assorted life-saving drugs, etc. Then incidence of severity of incidents are proportional to length of induction time in males, state of excitement. So that should be avoided. But besides that, in areas like Kaziranga National Park, where we work, we have another threat of running the animal into water. Okay, so we have to guard that side with the help of elephants. So we need number of elephants in this operation. Then we need the specific antidote against the narcotic, positioning for maximum ease of respiration, thorough inspection of the mouth. Sometimes even the tongue can cause obstruction to the respiration. Sharp pressure on rib cage if there is suspicious poisoning or overdoses, like that. So, because the translocation operation is a pre-planned operation, it is not a sudden thing. So everything is kept in boxes, and so on the previous day, the forest guards and every people will be gathered, and there may be a mock exercise, mock translocation operation. How whole operation will progress, we have to show them, so that the actual day of operation, there is no confusion. Now they are our very useful helicopters. In Africa, they use helicopters. In our case, the elephants are very useful. We use elephants for darting also. So like that, when everything is ready, we keep our tranquilizing gun and all that, we go with elephant. Then once the desired rhino is noticed, then everybody is alerted, communicated that yes, we have seen a very good young female rhino that we want to pick up, like that. Proper communication among different groups. Then the dirting. So for rhino, you have a very limited dirting size. The buttock if you are shooting from a distance, and neck muscle if you are closer, like this. So it doesn't work. Anyway, in the zoo conditions, I was trying to show this can be shot in the neck only from a short distance. If it is a long distance, you probably cannot do that. So the dart, you can see, dart has hit the animal. You may target here, but you have from long distance you cannot. So initially, within 4 to 5 minutes, the rhino will stand still. Then you will see that the hind quarter is dropped. Don't disturb the animal. Just look from a distance. Don't make any movement. If you make movement, the rhino will get disturbed. Then you will find that the rhino will lie down, and you may have a surprise visitor to inquire whatever could have happened to his auntie. Anyway, so then we go closer, we cover the eyes with a blindfold, as well as plug the air canal to block the auditory and visual stimulus. Then from behind, we use a stick or throw a pebble to see whether the animal is properly immobilized or not. Now, when the veterinarians progress with their procedure, like taking pulse, respiration, temperature, oxygen saturation, and all that continuously, you can see the ear plug and blindfold. The other group for like collection of the animals from their recovery, they also work in tandem. That is why everybody is kept abreast over what is going on. So like that, the JCB will come, slice an area adjacent to the rhino, and next they will, you see, the rhino, the work is in progress. Radio coloring. Now, there are different groups and every group respect each other. There is no shouting and everybody work in tandem. So, this is the slice. Once the area is sliced, right behind the animal, the JCB will pull the slice and place there. And then, this is the floor plan of the, what is called that slice, wooden slice. So, slicing of the area, the animal is manually turned over the slice, then it has to be carried to the wooden crate constructed by robust sal trees. So, the wooden crates are here where the animal is sliced by the JCB. It's a very heavy animal. Animal is 2000 kg, and this slice itself will be over 100 kg. Sorry. 1000 kg or 800 kg. Okay, so humanly it is not possible to drag. Moreover, you may have a muddy field or rough field, so you need that machine, what is called forklift or JCB. Now, this is the box. Two side doors are raised, and there is a ramp so that the animal can be, the slice can go like this. So once it will be up to here, then they will release, the JCB will go to the other side, from there it will drag. So now you can see the sledge is being dragged through the box. So once inside the box, the animal, so we partially drop both the sides of the doors, and then you can see only leaving the face side so that we can administer the reversal drug on the vein. And within 2 minutes, the animal will stand. And that is the time animal will struggle and be very aggressive. And that is why we not only give narcotic, we also mix with sedative. So if we are actually translocating an animal, we, along with atropine, we give acetylpromazine. Now, repeated acetylpromazine is not good because this can cause photosensitization. So, if we need to give further sedative, we may go for haloperidol, long-acting sedative, or azaperamide. So now intravenously the drug, reversal drug will be used, and then immediately the dose will be dropped, and then the whole box will be covered with agronet to protect from the heat. And around that time only you have to pour a lot of water so that the animal is cool. Hyperthermia will be there when the animal will be struggling. So like that. Then the crate will be lifted by crane and the lorry will be driven behind so that the cage is inside. And in this process, the face of the rhino has to be towards the front side because you have to go long. If the rhino is standing in the reverse direction, on transit it might get imbalanced or disbalanced. So that is why the head side is front. Now that creates a problem at the time of release. Because when you dig a ramp and try to lift the rear side, the rhino has the back side and it doesn't know that the back side is open and the rhino needs to go out. And a big animal, he cannot turn also. So what we do now, so we have experienced that in the initial stages of translocation. Later on, we have started another practice. So this was the first time where a ramp was dug and the rhino was released like this. We had to struggle for 15 minutes, 20 minutes before the animal could go out. Now we release like this. We lift the box and keep it in the ground. Then we open the front doors. Now you see two animals because they are mother and son. That is why we release them simultaneously so that they can find each other. But these animals have a habit. After you open the door, see, we are all safely up here because you cannot stay. The animal will kill you. It's a very violent at that time. Or even once this type of muscle was also threatened by the rhino, they go and hit there. Anyway, they come to the crane and start fighting. So they create a problem. Anyway, eventually, the people shout and blow whatever is there, and the rhino, they leave. They try to do some more damages for disturbing them and bringing them there. So I am talking about the Manas National Park, where we have a very successful translocation program, and all the translocated animals, even see mother, daughter, even third generation has been born there. Beautiful, pristine grassland, and now till now we have 44 rhinos. Not all are translocated. Okay, about 36 animals have been translocated. We have a lot of birds in captivity. We also had poaching. Initial stages, we had the problems of poaching, but the loopholes are being tried to block, and now they are better. And so, more or less, it is a successful operation. So, the whole operation credit actually the veterinarians can claim, because without the veterinary intervention, such a big operation cannot be carried out. And full responsibility is with the veterinarian. So it's a very successful operation that we can take credit. Now people take, because every animal is radio collared. So you know their movement and see like that. So with this, I want to thank, and if you have a question, we can take questions. Original is not related. So, sir, I conclude with this, so we can take questions. mic is off. Dr. Savarkar, I cannot hear you. Please unmute.
Sharma sir, just a minute, sir. The electricity had gone here, sir.
Okay, now I can hear you.
Yes, sir. Very wonderfully presented, sir.
Thank you very much, if you like it.
Sir, Dr. Savarkar is here. He is the chairman of this session. I will just request Savarkar sir to share his views.
Sure.
Sir, hello.
Yes, yes.
Very nicely presented, sir, and whatever the experiences you have shared. Now, regarding this rhino, I would like to invite students if they have any question.
Sir, can I ask a question? I'm Dr. Nayan Jacob from Pondicherry.
Dr. Jacob.
Yes, sir. Can I ask a question?
Yes.
Please go ahead.
Sir, I just want just two small doubts, sir. One, if the horn of the rhino is trimmed, what are the disadvantages that the rhino would face in its day-to-day life? And secondly, I would like to know about the social life of a rhino. Means how does it stay? Does it stay in a herd? Is it a single living? Is it matriarchal family? Or those things, sir? If you can kindly throw some light on that.
Yeah, I will try. I could not hear your first question. I will ask again, but I am talking about the social life of rhino. Actually,
the first question was about the trimming of a horn, sir. About trimming of the horn. If the horn of the rhino is trimmed, as you showed in one slide,
so first,
how is it?
I can hear you. Let me answer. So the social life of rhino is not like elephants. Elephants are very social. Generally, when the calf is born, the calf is very much attached with its mother, and till the second baby is born. Okay, so sometimes what we have observed, if the first baby was a male baby, the mother will still like to take it alongside, because the male babies are more vulnerable from either from predators or from other male rhinos. Females are generally not as vulnerable as that of the males. They don't have very good big social groups, but sometimes in a field you will find that there are 10, 12 rhinos grazing together. But they are not a family group or anything. They come together by chance, but they again go back to their respective areas. They are not very territorial. To some extent, they are territorial. Okay. Then regarding the trimming of horn, the rhino horn is not a true horn. It is only a hair follicle, one hair follicle that grows and takes the shape of a horn, and it is not as hard as buffalo horn or cattle horn. It is very soft. It can easily be removed. Now, if you trim the horn, just like our cow horn, it regrows, and physiologically, it doesn't have any significance, because some people believe that only a male with a very beautiful horn is accepted by the female, and he is attractive to the female. It's not true. A rhino can reproduce without horn also. Horn is not a reproductive organ. And some people also believe that the mothers need the horn to guide its baby or to protect itself from predators. That is also not true. Actually, in case of rhino, the lower incisor is the main offensive and defensive organ, lower incisor, which is very sharp and very strong, not the horn. So horn growth, I don't know, but they can do quite okay without the horn. So if you undertake a project of trimming, you can very well increase their chances of survival. Am I clear, sir?
Thank you, sir. Thank you for your answers. Thank you very much.
Sir, you have referred regarding electronic surveillance. So can you share some experience regarding GPS tracking system if you are using?
Yes, yes. In the radio collar, actually we use in case of rhino the satellite collar system. So we have absolutely no problem with that. The problem with the radio collar is that sometimes even after the signals are dialed down, the radio collar do not drop. We prefer to use an auto drop type of radio collar. There are two types of auto drop radio collar. One is which is biodegradable, and because of the degradation of the some part of the radio collar, the collar should drop maybe after 3 years. But quite often we have seen that it doesn't drop, and there is an auto burst type of design radio collar, which is very expensive. We have not used that. Like after a certain period, there will be a small burst so that it will dislodge. But we didn't use that type of collar. And we have seen on a few occasions, the collar has cut deep into the neck of a growing animal, and we had to go and recapture that elephant for removing the radio collar. Sometimes it causes a problem. So we do not prefer to use a radio collar in a growing animal. In fully grown animal only, we place the radio collar. Otherwise, the signals and all are very good.
Sir, we have observed at the Gir National Park regarding the radio collar on the lions, the adults also even are found to have some behavioral changes due to the radio collar, and it disturbs the animals in hunting process also. So
yes, sir. Yes. In case of lion, in case of tiger, the radio collar is supposed to. People say that they create problem in their hunting and like that. I have not experienced, because here we have not radio collar tiger. We don't have rhino in our, because you don't give. If you give, we want to bring some lion from Gir. Just a joke. Our territory is not suitable. We have enough already. So anyway.
Sir, when I have visited the Kaziranga National Park, I have seen the forest guards are also having the automated weapons for guarding the rhinos and tigers and all that.
Yes.
Whereas in Gir National Park, we are having no problem of the poaching. So our guards are only carrying the lathis or sticks with them, and only foresters and above ranks are allowed to have pistols or revolvers with them, firearms with them.
Yes,
so
they use those weapons for self-defense. By chance, the lion may attack and they need to protect themselves. In case of Kaziranga, the whole situation is totally different, because you know we have poachers from China, neighboring country. We have people from Myanmar, and you know this is one rhino horn costing more than 1 crore. It's more than a lottery if you can get one rhino horn. So that is why there are international investors, and we our people, our forest guards, they have to fight against those international these missionaries. So that is the big problem. And you know, even we have heard the Chinese people come and they sit in Nagaland, they marry a Naga lady, and then and they stay here and they invest money from in Manipur also, from Surchanpur district. We know all those linkages. but it is extremely difficult for Assam police to go and arrest them because neighboring state there are problems. They may have political support also because it is a big money.
Yes,
So
sir.
the only thing that we can do is shoot at sight. So in Assam,
Okay, sir.
in a national park or in Manas National Park, so whenever they see a poacher or intruder, illegal intruder, they can shoot. So that is why, see, last year we had only 2 poaching. A couple of years we were having very high number of poachings. Now it has come down. Of course, the BJP government in Assam is also, that was their whole promise last time, that we will protect the rhinos. They are the pride of Assam. So they were successful.
Great, sir. Great. Sir, one more question I would like to ask you. If you want to collect the blood from the rhino, then from which side it is easier to collect the blood?
Ear vein.
Ear vein.
Ear vein is very prominent.
Okay, sir.
We do it there only. Blood collection or giving intravenous medications like that. Ear veins are very prominent.
Okay, sir.
Okay, sir. Thank you very much for your nice presentation and share very good, valuable information for our students. So, on behalf of organizer Dr. Anshu Elawar, Associate Professor and Head, Dr. Udit sir. So, again, we are really thankful on behalf of organizer, college and university. Thank you very much. Thank you very much, sir, for sparing your valuable time.
Sir, to everyone and your dean, my good friend, Professor P.H. Stan. Please convey my thanks and to all of you for considering me to deliver some talks in your very important program. So we enjoyed and some of our students also joined us. So they have also probably enjoyed. Did you enjoy? Yes, sir. Okay. They said they have enjoyed. So anyway, thank you very much and like in future also if you think there are some such program and you can always count on me. Thank you, sir.
Sir, I would like to remind you one thing. Sir, I would like to remind you one thing. We have met at the Jaipur conference of the LPM. And we have got your photographs also. We have got photograph with you also, sir. And that is the lifetime remembrance. And hope that we will meet again in some near future in the conferences of LPM or surgery, sir.
Okay, sure. Let the go. I will come to Junagadh.
Welcome, sir. Welcome.
Okay, thank you, sir.
Okay, thank you, sir. Thank you very much, dear participant, student, trainees and friends for joining with us since morning. So thank you very much. So tomorrow we will meet at 9 o'clock. Thank you.